So, so welcome to our up to speed session number 2. Okay, welcome to up to speed session. So, these are going to be your rapid catch up sessions. If you are going to be joining our regular theory class, then these are your best, let's say, best option with minimum time wastage that you can cover everything that we have done up to this point in our theory classes. Okay, so that's why I especially started this. Okay, so if you are an online student, you have two options of joining with our normal theory class. We have a Saturday batch and a Thursday batch. Okay, so all the information is available in our groups. Okay, so hopefully everyone that is joining this meeting are in our groups. Okay, so if you are not, you can send me a personal message in the Zoom, then you will be added. Okay, so if there are new people who are joining for the first time for a live up to speed session, okay, so welcome. And this is, as I said, these sessions are mainly focused on teaching you what you missed in the most time saving manner as possible. Then we are going to complete the tutes together. Okay, so tute completion is going to happen, as well as sort of a I'm going to bring you up to speed, as the name says. Okay, so that is what we did last week. We have covered up to this point. We have covered up to we started the second lesson, right? We finished introduction to biology. Okay, we finished introduction to biology, and then we started our second lesson. And in the second lesson, okay, in our second lesson, we finished the water part. Then we have to start with the organic molecules of the biological molecules, that is carbohydrates. Sorry, the carbohydrates, lipids, proteins, and nucleic acids. I'll try to finish today. Okay, so let's see. Okay, that depends on our cooperation, right? You have to keep the chat active, keep the main chat active, because that's what I have open right now. So keep the main chat active. You can ask any question, any doubts. The tute is available in the Zoom chat. The tute is available in the Zoom chat. Please download it. But I think if you are enrolled to our website, if you are an online student, then I think you must have received delivery of the tute. If you have enrolled to either our Thursday batch or Saturday batch, whatever, you must have received delivery of 3 tutes. Our first, the blue color one, and then our second main tute. I think both were delivered. If they were not delivered, they probably are on the way. If you enroll to the website, Guru Tharu Online website enroll, it should be on the way. Okay, and if you're a physical student, of course, I think I handed you the tutes in person. All the physical students must have got it. If you somehow, if you're a brand new one, right, fresh, you can download the PDF for now. As soon as you get your hands on the tute, you can complete it. This recording will be sent to the groups. You can ask any question from my team. Okay, I have 2 hotline numbers that are almost always active. Okay, then you can drop them a message, you can call them, they will call you to get an update. Okay, so everything is there so that you are not left behind. So from the beginning, you are going to be guided, and just it's a journey that we are going to take together, right? As the teacher and the student, and right, that is going to go ahead. Any issue you can have, you can ask during the class, or you can so any issues up till now? Yeah, you can ask anything in the main chat. Okay. The PDF shared on. Okay. Okay. Please add me to the group. Okay. Will be added. Okay. Send me the number, please. Without the number, I can't add. Then the PDF shared in the group. Yeah. That's the same thing. But I think I added. I think I sent my annotated PDF again. The PDF I wrote on. So because some students requested that one as well. The PDF I sort of did all these edits. I think I sent that one when I make it, right? That is also there in the group, and I will send this clean PDF that is available in the Zoom chat. Okay, so download it from there. I will send it again. Okay, so as I said, we finish introduction to biology. We finish introduction to biology with a nice discussion of all these issues. and our tute was completed. And if you didn't join, you can watch that recording. Okay, but only one up to speed session was done. So if you're joining today, then good, right? Nothing to worry about. Okay, so we will. So we took the start into the second lesson, and we discussed about water. And there's a water essay, ne? There's a water essay that needs to be marked, but I think I sent the marking scheme. Yes, hands up if you saw the marking scheme. Water essay marking scheme. If you saw the marking scheme, I gave you an essay. What was the essay topic? Essay topic essay ka clear na. Yes. What was the essay I gave you to write? Yes. What was the essay I gave you to write? Chemical and physical properties of water vital for life. Yeah, that's what I gave. That's what I gave, right? So the marking scheme is available in the groups. I'll be resending them. Okay, no need to worry. And okay, so we'll take the start then, right? Anything you want to clarify? Anything else you want to clarify? Okay, so this is an interactive session, as you might have seen if you have already joined the theory class, right? Main thing in bio is that during the theory class you have to pay attention and just get the sort of the extract what of the lesson that is being done today. If you are learning about carbohydrates today, just have a nice summary in your head, so that when you are doing your self study or when you are doing questions with me, you have a sort of a outline. That's enough. That's the first thing you need to do in any first time you are learning anything. That's it. Then again and again when we are doing papers, we have a exam coming up. So I mean, after these up to speed sessions are done, then I'll be having an exam for online physical everyone. Okay, so in that exam you can perform. Okay, no pressure, it's going to be a small exam, right? Just so that you can have a small flavor of what is A level biology and also how my class is structured. Okay, so my class, I try to have a balance between keeping your interest as well as guiding you to the best result. Right? So that's what I'm trying to do. I try to keep it energetic. I try to keep your interest as well as I get the work done. Okay, so those are the principles. So we'll have a chat while we are going on. It's already 6:27, so we'll start. Okay, any questions? Okay, I'll come to the essay discussion. Okay, I'll come to the essay discussion. How to write an essay, and also I'll go through the marking scheme I gave you that is about water. So I'll come there. But we'll take the start. No, we'll take our today's discussion. So we'll start with carbohydrates. Okay, so we'll there's going to be no note for you. We are going to just take down some things in the tute, and then we are going to do questions. That's how my classes are structured. Okay, if you go to, so this is not how my ordinary tute looks like. If you have received my second tute, that is how my normal tutes looks like. It is only diagrams. Then there's another tute with only questions. That's all. Okay, then there's a separate book to write essays. That's all I'm doing. Okay, every week we are trying to write essays. Every week I'm going to mark essays with you, or I'm going to give you marking schemes. Okay, carbohydrates. Let's read. I want you to highlight. Okay, highlighting is a very good visualization technique. Okay, highlighting helps you to see things that are hidden in the textbook. Okay, so have a nice set of highlighters. Then get a nice pencil book, pencil case, and maybe steal some highlighters from your, if you have sisters or someone, or if you can just buy some good highlighters. Okay, because visualization is very important. That's why I put color diagrams in my tubes, everything. Okay, biology, try to get it into your mind's eye. Got it? Okay, some theory points. So keep a color allocated for definitions. Okay, what do you mean by definitions? That means sentences that are describing exactly what something is that you find in the biology syllabus. For that, keep a color separate, and for other things, highlighting some general points, keep some other colors, very simple colors. Okay, so something like this. Most abundant group of organic compounds on earth is carbohydrate. Most abundant is carbohydrates. Highlight that. That's a theory point. That's a theory point. Elemental composition is carbon, hydrogen, and oxygen. Okay, that's why they are called as hydrates of carbon. They are hydrated carbon sources. That means what? Hydrated means you have added water. Okay, to carbon containing molecules you have added water. So that, why do we say there is water in carbohydrates? Because you find hydrogen and oxygen in the same ratio as water. What is the ratio of H2O in water? H2 oxygen in water, that is 2 to 1. Yes. So that's what they are saying. Okay, then there's a general formula. There's a general formula. That means you can write any carbohydrate on earth using this general formula. So fill in the blanks. It is CxH2O within brackets Y. Okay, CxH2O within brackets Y. Got it? Okay. Three major groups of carbohydrates are monosaccharides, disaccharides, and polysaccharides. Okay, highlight that. The three major groups of carbohydrates are monosaccharides, disaccharides, and polysaccharides. Carbohydrates include sugars. Okay, these are sugars. Okay, monosaccharides and disaccharides are considered as sugars. මෙහෙම draw this sort of මොකද මේකට කියන්නේ වෙන මෙහෙම that shape, draw that and label them as sugars and polysaccharides. Okay, clear? So what I want you to do is, okay, here, don't come up to, when may keep a small mental separation here. In this space, in the tute, in this space, I hope you have found out which page we are doing. We are on page number 11. Okay, we are on page number 11. Page number 11 again. Okay, so I want you to do a small classification, right? Carbohydrates, you can classify them as sugars and polysaccharides. Are the okay, sugars and polysaccharides. Under sugars, you have monosaccharides and disaccharides. Okay, under sugars, you have monosaccharides and disaccharides. Understood? Do that classification quickly. Carbohydrates, you can classify them as sugars and polysaccharides. Okay. Okay. Yes, of course, I provide recordings. These are free sessions done as a special favor for everyone who's joining new. Okay, yeah, the recording is there, but try to stay live. Okay, otherwise, I know recording it's going to be around a 3.5 hour recording. So I know you're not going to go through the entire recording. Maybe you speed me up, right? 1.5 data. The tute is not downloading. Anyone who else is facing this issue? Tute is not downloading. Anyone else facing this issue? Hands up. The tute is not downloading. Anyone else? Okay, I'll send to the group. Okay, check now. It's available in our main announcement group. People who are not on the main announcement group, please get added. Not for physical students, online students only. Okay, may get added to the our announcement group as well if you are in other small groups. Okay, okay, yeah. I sent to the group. Okay, all right. Moving on. So we'll get on to the discussion of what are monosaccharides, right? So simplest form of carbohydrates having general molecular formula. So you know under carbohydrates you have for all carbohydrates you can apply this general formula. That means the carbon number can be X, right? It is variable. The hydrate or the water molecule number can vary, that is Y. It's a varying number, but hydrogen to oxygen ratio is always going to be 2 is to 1. Can you see that? Hydrogen to oxygen ratio is always going to be 2 is to 1. You will see this difference you will see when we are moving on to lipids. In lipids, this ratio is not there, hydrogen to oxygen. Okay, that's why I am highlighting this. They contain the same proportion of H2O, which is equal to 2 is to 1. Okay. Then write down there's a separate general formula for monosaccharides. I mean, it is not a separate general formula, but it's something that we derive from this. Again, from this main general formula, we derive another general formula for just for monosaccharides. Okay, what is the just for monosaccharide general formula? It is CH2O, the whole thing within brackets N. Okay, got it? Quickly write that down. That means carbon to hydrogen to oxygen ratio is going to be N, 2N to N. Got it? So the same number of carbon is going to be there with oxygen, but hydrogen is always going to be 2 times. Okay, that is the general formula of monosaccharides. The carbon number varies from 3 to 7. Carbon number varies from 3 to 7. Jump here, Metin. Can you jump here? So I have given all the carbon, I mean varying carbon number monosaccharides I have given. Okay. So if you have a 3 carbon monosaccharides that you can call as a triose. Okay. You need to remember the examples for the triose. You need to remember the example for the triose, that is glyceraldehyde. Okay. Then tetrose is 4, right? Erythrose. I'll explain again once all these molecules are. Pentoses, that means 5 carbon atoms. That means you have ribose, deoxyribose as well as ribulose. 6 carbon are the most common. They are called hexoses. Hex means 6, no? Right? Glucose, fructose, and galactose. Okay. So the 7, so we say here carbon number varies from 3 to 7. No? You don't need to know the 7. Okay. Heptoses you don't need to know. Hexoses is old enough, right? Because those carbon 7 containing molecules are very rare. Okay. No use. As they are not mentioned in your syllabus, no need to know actually. Okay. Got it? Okay, hurry. Api will write down some simple properties of monosaccharides. Okay, I think you had in O levels as well. These monosaccharides, disaccharides. Right? O levels you also had no? Basic carbohydrate knowledge. Right? Don't overthink about anything. Just follow what I do. Okay, then with time this will all come to you. Don't overthink. The main thing I see in my 2028 new batch compared to maybe previous years, you guys overthink. Right? You overthink everything. Okay, small hidden details. Okay, but yes, it's good to be curious, right? I will always explain small things again and again, but don't overthink. Okay, don't overthink. Just focus on the key points and then just go with whatever that is happening. It will all come to you in due time. Everything comes with a little bit of experience. Okay, what is A level? How do you take notes in A level? How do you write an essay? How do you use your resource book? Everything comes with experience. Okay, so don't worry too much, don't overthink too much, right? Particularly the subject of bio is not as difficult as they say. Maybe physics could be difficult, chemistry could be difficult. For me personally, even though I'm teaching bio, my favorite was chemistry, then bio, finally physics. Okay, so biology, relatively, it's a easy, stress-free subject to learn. Okay, but it can be a bit difficult to study. But my advice to you is, okay, don't overthink at this point. Okay, try to gather knowledge, try to gather experience, listen and pay attention. Okay, be curious, ask questions, of course, but don't worry too much, don't get stressed too much. Got it? Right, properties of monosaccharides you need to write down here. Let's write down what are the properties of monosaccharides. Okay, they are all considered as sugars. Therefore, you can say they have a sweet taste, but let's write it at the bottom, right? So, they are all water soluble. Got it? You can dissolve all your monosaccharides in water. So, they are water soluble. Either they are water soluble, got it? You can dissolve them in water. Then they are crystalline, no? Have you seen? So, what are the examples here? Have you seen glucose powder? If you zoom into a glucose crystal, you can see it is having a small microscopic crystal-like shape. Okay, so it is crystalline. Got it? Then uh, there's another concept. You can just write this down. I'll explain. They are reducing sugars. They are reducing sugars. Got it? They are reducing sugars. Okay. Write these three down. They are water soluble, crystalline, and they are reducing sugars. Those are the properties of monosaccharides. Okay. Yeah, of course. As I mentioned, the recording will be sent. Okay. So if you have any connection issue, don't worry about it too much, but join the live as much as you can. The recording will be sent to all the groups, as I did last week. Okay. Okay. Right. Clear about our 3 to 7 carbon number as well as our general properties. Okay. Then what are these? Our trioses, you have glyceraldehyde. Okay. Glyceraldehyde is a 3 carbon containing monosaccharide that you will encounter when we are doing, actually the lesson that we are currently doing in the main theory class, that is photosynthesis. We will very soon do this in the Kelvin cycle. It is upcoming this week. Okay. Then erythrose is very rare, as they say, so you don't need any applications or any examples. You don't need to know. Ribose and deoxyribose, we'll learn about it very soon in DNA and RNA. Okay, can you draw an arrow to ribose? Can you draw an arrow to ribose? In RNA, you know RNA, no? RNA, ribonucleic acid. We'll learn within today if we have time. Then draw an arrow to deoxyribose, right? In DNA. Everybody knows DNA, which is how we store our genetic information. Got it? Okay. Hexoses. Hexoses, of course, these things you know. No? Glucose is our main energy source. Fructose is found in where? Can anyone tell me where fructose do you have? What is the commonest example for fructose? Yeah. And galactose. Yeah. Fructose is found in. Yeah. What happened to the chat? Fructose? Where do you find fructose? Yeah. Okay. Inulin. Inulin. Where else? Where else do you have fructose? In honey, no? In bee honey? Yeah. In ripe fruits you have. In bee honey you have fructose. Yeah. That's why fructose can be considered as one of the most sweetest sugars there is. Okay. So bee honey you have fructose. Okay, fruits, particularly fruits that are about to get ripe. I mean, very close to ripening. Yeah, mangoes. Have you seen very yellow color mangoes? Right, the sort of very ripe fruits have a lot of fructose. Then something, this is as I keep on telling, as a bio sir, I have to teach chemistry to you guys first. In the enzymes, I have to I had to teach you about reaction kinetics, reaction mechanisms. I had to teach. Now I have to teach you organic chemistry. Okay, according to the type of carbonyl group, okay, they are classified as aldoses and ketoses. Please highlight this. According to their carbonyl group, okay, they are classified as aldoses, ketoses. Okay, I have given you what an aldehyde group is. I have given you what an aldehyde group is. Got it? What can you see here? Yeah, what can you see here? How many carbons do you see? 1, 2, 3, 4, 5, 6. That is because this is an error in your resource. Okay, I yeah, it's given here. Okay, if you have a carbon here, can you please correct it to a hydrogen? Please correct it to a hydrogen if you have a hydrogen here. Sorry, a carbon here. Can you see? Here, if you have a carbon here, please correct it to a hydrogen. Okay. An aldehyde group, listen, an aldehyde group is, if you have a chain of carbon, if you have a chain of carbon like this, at the end of that, if you have a double bond oxygen and a hydrogen, this group is called as a aldehyde group. A double bond oxygen and a hydrogen, if you have, that is called an aldehyde group. Okay. So those are aldoses. If you have an aldehyde group, that is called as a aldose sugar. Then what do you mean by the ketose? Keto group is this. A keto group, listen carefully. A keto group, if you have a chain of carbon like this, right? In this carbon that you have here, there's a carbon with a double bond oxygen. So on the other sides, you have two more, on the other sides, you have carbons. There's a double bond oxygen that is called as the keto group. Can you see here? So if you have an aldehyde, if you have an aldose, if you have an aldehyde group, you are known as a aldose sugar. What are the examples for aldose sugars? You need to remember these: glucose and galactose. Highlight. The examples for ketose that you need to remember is fructose. Okay. Why is it a ketose? That is because he has a carbon double bond oxygen like this. Okay. Then highlight this. You don't need to remember. Okay. I gave this in my tutor as well. You don't need to remember these chemical structures. This is not organic chemistry. Got it? This is not organic chemistry. You don't need to remember these structures. Okay. Then in aqueous form, when you dissolve glucose, when you dissolve fructose in water, they are going to sort of, this is called the linear form. This is called the linear form. That linear form is going to fold it into a ring-like shape so that it exists in this ring-like shape in the aquatic medium. Okay. Got it? Okay. All right. Then let's move on to disaccharides. Okay. They are sugars formed by joining two monosaccharides by a glycosidic bond. Okay. I have to explain what is reducing sugars. I will come there. Okay. If I forget, remind me. Harry. Okay. Come to disaccharides. They are sugars formed by joining two monosaccharides by a glycosidic bond. Okay. So a glycosidic bond I have given here. How do you make a disaccharide? You can write down here. How do you make a disaccharide is, you take a monosaccharide like this and you take another monosaccharide like this. Then you join them. By a special bond. What do you call that bond? What do you call that bond? Yeah, a glycosidic bond. Glycosidic bond. You take two monosaccharides, you connect them by a glycosidic bond, which is a type of bond that we'll talk about. Then you have a two monosaccharide containing substance that is called a disaccharide. You had this in O levels also. A glycosidic bond is formed by the removal of a water molecule from the two adjacent monosaccharides by a condensation reaction. Please highlight this. There is a condensation reaction happening when you are forming a glycosidic bond. The water molecule is formed from OH group of one monosaccharide and hydrogen from the adjoining monosaccharide. Highlight OH and H. Those are the two groups that are getting involved when you are releasing this water. So I have given how it is formed. So you know how disaccharides are formed, right? I have given you two examples for disaccharides here. If you connect two glucose, you are going to get maltose. If you connect two glucose, you are going to get maltose. If you connect glucose and fructose, you are going to get sucrose. You are going to get sucrose. So this is where you have the glycosidic bond forming. Can you see? So this monosaccharide is giving an OH group. This guy is giving a hydrogen only. Then they are going to combine, and at the end, what's going to happen? There's something. Can you see? There's a water molecule that is being released. So if something is losing water, for example, if your body is losing water, what do you call that? If you're losing water, what's going to happen to you? What do you call that? You're going to get dehydrated. Yes. Yeah. You're going to get dehydrated. If two molecules are losing water, they are also going to get dehydrated. So this is a dehydration reaction, or you can call it a condensation reaction. Okay. So can you write here? Okay. Can you write here? Draw an arrow, glucose and glucose plus sign, put a plus sign, and then you can write here, it's a draw an arrow and write on top condensation slash, it's a dehydration reaction. Okay. And I want you to highlight nicely this bond. It's a glycosidic bond. Okay. Glycosidic bond. Similarly, the same thing is going to happen. A condensation or a dehydration reaction is going to happen between glucose and fructose. You are going to get a nice glycosidic linkage. Okay. Still we are in O levels. Okay. Right. These are easy things to don't need to overthink at all. Okay. The molecules you need to overthink are proteins and maybe a bit of nucleic acids. I'll show them using the 3D software and everything. Yeah, of course, and that's a good question. Ask those kind of questions. Okay. There's a question: can you break down a disaccharide into monosaccharide by adding water? Yes. Okay. In my practical seminar, I think it's available in my YouTube channel. Okay, I'll send that to you once I complete all these biological molecules. Okay. There's an experiment where I use sucrose. To sucrose, listen. Okay. To sucrose, I add a dilute acid and I heat it. A dilute acid, HCl. Okay. In organic chemistry, you will learn that if you want to add water, what you do is you add H plus in an aqueous medium. Again, H plus. That means H plus means what? It's an acid. No? Then if you heat this, you can break down the sucrose into glucose plus fructose. Right? In organic chemistry, you will do this hydration. We are going to hydrate this molecule and we are going to break it into two. So if you do this is the same reaction can be done in the opposite way. This way, you can add water to sucrose and you can break it into the two monosaccharides. Okay, you can add water to the sucrose and you can break it into the two monosaccharides. Okay. Okay. So we'll write down the properties of disaccharides. Let's write down the properties of disaccharides here. Okay. No need to write this. Okay. I just wrote it as an example. Okay. No need to write this. Let's write properties of disaccharides. Number one, water soluble. Number two, crystalline. Okay. Crystalline. Then I want you to write down a very special point. Some are reducing sugars. Some are reducing sugars. What are the reducing sugars out of the three disaccharides? Out of the three disaccharides, Listen, okay, maltose and lactose are reducing sugars. I'll explain what is reducing sugars within a minute, okay? Then I want you to put a star mark here. Sucrose is not a reducing sugar. Sucrose is not a reducing sugar. Can you quickly write this down? Some are reducing sugars, maltose and lactose. Yeah, you can call it as hydrolysis as well, the previous reaction. When you are breaking down a disaccharide into monosaccharides, you can call it as hydrolysis as well. You are lysing using water. You are separating, breaking using water. Quickly write this down. Okay. Okay. Yeah, there's another disaccharide missing here. There's a disaccharide missing here. After you have written this, I want you to add. So there's only, I have given you only maltose and sucrose. No, I want you to add the missing disaccharide. Can anyone tell me what is the missing disaccharide here? Yeah, what is the missing disaccharide that I need to add? The other disaccharide that you have in your syllabus, O level salt you had. Yeah, it is glucose plus galactose. You are going to get your milk sugar, lactose. Yeah, you are going to get your milk sugar, lactose. Lactose. Add it here as well. Okay. Maltose, sucrose, and lactose are the three disaccharides that you need to know. Then their properties are these: water soluble, crystalline, some are reducing sugars. Okay. Then listen quickly. I'll explain what do you mean by reducing sugars. I told you I have to do chemistry, teach chemistry as well. Okay, in chemistry very soon you will, in the third lesson you will do something called redox reactions. You will do something called redox reactions. Don't overthink about this too much. Okay, you will learn this in chemistry. Okay, imagine you have a molecule. This is some molecule. Got it? If that molecule, listen, if that molecule is giving electrons, losing electrons, what do you call that process? If a molecule is giving electrons, that we call that as oxidation. You are losing electrons. Then there's another guy. Someone. There's another molecule here. That molecule is accepting electrons. That molecule is accepting this electron that was given by Minamaya. Got it? So we say if someone is accepting electrons, that is called reduction. Reduction. Okay? If someone is giving electrons, that is called oxidation. If someone is accepting electrons, that is called reduction. Okay? So then the word agent or reducing sugar. Okay? So imagine if this red color thing is a sugar. What do you mean by a sugar? Yeah, if this is a sugar, what do you know as sugars? Sugars are either monosaccharides or disaccharides. Sugars are all only monosaccharides or disaccharides. So when you call someone a reducing sugar, that means he's reducing someone else. He's reducing someone else. Okay, he's reducing someone else. That means because of him, someone else is undergoing reduction. Okay, someone else is undergoing reduction. Okay, so can you tell me? So in reality, okay, actually, the reducing sugar is undergoing oxidation. He's reducing someone else. That's why he's a reducing person. So actually you are reducing sugars are undergoing oxidation, they are losing electrons, they are giving out electrons. So when they are giving out electrons, they are undergoing oxidation. Someone else is undergoing reduction, accepting. Then in these reactions, okay, nobody is giving electrons if no one is accepting. No one is going to give electrons. This guy is giving electrons because there is someone to accept. Got it? He is undergoing oxidation because there is someone to undergo reduction. Okay? So, kawuruth electron එක ගන්න නැත්නම් he is not going to give electrons, right? That is not going to happen. Just oxidation is not going to happen. Okay? So, you can say this reducing sugar is giving electrons and someone else is accepting. That is something we test in a practical. We have a practical to test for reducing sugars. We have a reducing sugar practical. Okay, those practicals are covered in the practical seminar. There's a very nice video in my YouTube channel. Okay, we'll discuss that theory once more at the end of this session when I'm going through the practicals in a rapid way. But I recommend that you watch the full video where I demonstrate. I do the practical in the class and I show everyone the result. Got it? Okay, now understood why do we call these reducing sugars? Because they give electrons. So, Taggalakian, who's the only non-reducing sugar that you need to know? Who's the only non-reducing? Taggalakian? Who's the only non-reducing sugar? Oh, good. That's it. Okay. My point is, I don't want to waste a lot of time here because in your syllabus, when you go ahead, there are so many much more complicated things for you to learn. So those things, when you are learning those things, you will laugh at these things. Right? So in sometimes when you were small, your toys. If you wanted a certain toy, that was the only thing you wanted, right? So if someone told you in a certain some time, you will not even think about that toy. No, you are not going to accept that. So at this point, you are thinking, okay, these are the lessons, these are things that are difficult. But when you learn something like the female reproductive system, genetics, molecular biology, recombinant, and mutations, all those things once you learn, then you will even forget that these lessons were there. But you can't do that because questions are asked from these in the A-level paper. As we'll do other past papers, we'll do past paper questions within today. Okay, right? So that's it. Properties: water soluble, crystalline. Some are reducing sugars. Got it? We'll move on to polysaccharides. Everyone not sleeping, right? Okay, it's only 1 hour, not 1 hour, only 30 minutes into our session. Okay. Polysaccharides. Nice set of words are there. Please highlight this. They are macromolecules and biopolymers. Highlight. They are macromolecules and biopolymers, right? Simply, macromolecules are molecules with a large molecular weight. Okay, you can say, you can draw an arrow. They have a large molecular weight or molecular mass. Macromolecules have a large molecular mass, molecular weight. Why are they called as biopolymers? You know polymers, right? You know polyethylene, Teflon, or in O-level syllabus at the end of that you had, right? Teflon, polystyrene, all those polymers you had, right? So, what is the common thing about how does a polymer form? It's like this. Can you see all these polymers that I have given with you? All these are monomers joining together. So, what are those monomers? Those monomers are actually monosaccharides. So, the monomer of polysaccharides are monosaccharides. Okay, that's what we say here. Polysaccharides are made up of few hundred to few thousand monosaccharide subunits. I want you to highlight this: monosaccharide subunits. Monosaccharide subunits. Okay, then we'll write. Okay, let me know if I'm going too fast. Okay, you can ask anything. Let's write down some properties of polysaccharides. Okay, You can say water insoluble. You can't dissolve them in water. Have you seen wheat flour, palm pity? Can you dissolve it in water? It's going to so exist as like a sort of a floating clump, no? Floating mass of wheat flour. It's not going to properly dissolve, right? Water insoluble. Sorry, water insoluble. Water insoluble. Number two, okay. Water insoluble. Non-crystalline. Second one, write down. Non-crystalline. They are not crystalline, right? If you take a kilo of wheat flour, pump it from your mother's kitchen. If you take some wheat flour, wheat flour, rice flour, atta flour, whatever, okay, it's powder, right? It's not the consistency of a crystal. Sugar crystals, it's not like that. Non-crystalline, Harida. Final one of the most important ones, third one, not considered as sugars. Not considered as sugars. Not considered as sugars. Okay. Not considered as sugars. Water insoluble, non crystalline, and not considered as sugars. Okay. Polysaccharides. So, how's the A level experience so far? Frustrating, complicated, confused? Any idea? A level so far? Interesting? Yeah. Confusing? Okay. Complicated? Mostly confused? Okay. It's no use saying to a scared person, don't be scared, right? So I'm saying to a confused people, don't be confused, right? Because at this point, it's okay to be confused, interested plus confused. At this point, you can be confused, but in another year's time, if you are confused, then that's a problem. So if my 27 people are confused, then right, then it's something wrong with me or something wrong with them. Okay, because I have to take responsibility as well. So it's be confused at this point. Okay, what I say is you can explore, you can go to classes, you can join online to free sessions, other bio classes, other chemistry classes, other physics classes, you can go. Okay, even I am in several other biology classes groups. So whenever they are having a free session, I also join. If I'm driving and if I'm feeling sleepy listening to music, I listen. I go to another bio class free session. Then I sort of listen. I'm not trying to sort of find criticisms in them. What I'm trying to do is I'm trying to improve my teaching. Okay, because at the end of the day, what I should give you is all the good things without any of the negatives. Okay, so I go to other classes, I listen to other recordings, I watch YouTube recordings. Okay, so similarly, you should explore, you should understand what is the real goal of A level biology. Okay, you should understand that is to go to the paper and write a nice answer and come. That's it. But in that journey, you should not fall asleep in the class. You should not be scared if the person is not going to cover the syllabus. Then you should not be bothered if the class is deviating from the main goal, that is teaching you the syllabus. But at the same time, too serious, if they are just reading the resource book, then that's not also going to be useful. Okay. If polysaccharides are coming from disaccharides, why are they not showing similar properties of disaccharides or monosaccharides? For example, if you take a single brick, Gadolaggan, if you take a single brick, it has some properties. No, you can throw it. You can hit someone in the head with it. Yes, you can break it down into small things. It's just a single brick. If you build a wall with several thousand of those bricks, you build a wall. it's going to have different properties. No, it's going to be like a barrier. You can't go through with it. You can't move it. Right? When things get together, they have different properties. Okay. Complicated and complex. Local is harder than Excel. That's kind of true. Yes. Don't know how to start. Start today. Still not on the track. Losing my mind. Okay. Am I? Okay. Yeah. Okay. Pressuring to choose another stream. That's another thing. Okay. Um, if you are still confused between maths and bio, you need to do two things. Listen, if you are still confused between maths and bio definitely can just forget about this. If you want to become a doctor or a molecular biology scientist or a biomedical engineer, whatever, do bio. Okay, much easier than doing maths and becoming a biomedical engineer. Okay, combined maths is difficult. That's what I have to say. Combined maths is a difficult subject. You need to always pay attention. But whenever you are deciding on doing maths or bio, think of your goal, not just the subject. If it's maths or bio, don't think about that. Think if you are visualizing yourself as someone in the biological field. If you are willing to be a scientist, molecular biologist, dentist, veterinary surgeon, a doctor, a consultant, a surgeon, eye surgeon, cardiologist, endocrinologist, cancer surgeon, and whatever, visualize who you want to be. Then if it's something in the biological stream, whether you like it or not, you have to do the subject. If you visualize yourself as an engineer, someone who's uh, in the other fields that you can go by doing bio, for computer science, software engineering, whatever, then you should have in your mind the logical idea. Okay, I need to do maths to get to my goal. So if your goal is something that is not achievable by doing bio, then don't do bio. At the end of the day, it's up to you. I mean, it's all about who you want to be, your career, right, as a professional. Because I had no doubt in my mind when I started doing bio, I wanted to be a doctor. Okay, so I had the goal. Even though I didn't like biology much, I knew I didn't have an option. Okay, so if your parents are listening right now, this is a good time for you to have a discussion with your parents. Tell them what you want to be, what your passion is. At this point, you're almost 18 years old, and you can take a decision for yourself. At 18 years, other country people, they're almost starting their undergraduate degrees. Okay, we are doing A levels, right? Why we need physics to become a dentist or a doctor? That is because physics uh, builds our thinking. Physics builds our thinking. Okay, physics is really important. Otherwise, we are just going to be some memorizing people. Okay, physics is important to improve your thinking. That the brain capacity you build with physics is important for chemistry as well as bio. Right? You develop, you sort of you become smarter through physics. Got it? You become smarter. You try to apply. You apply equations. You apply a different equation. Right? You become smarter through physics. That smartness, that increased brain capacity, you can apply for chemistry and then bio. Okay. Can you give us a free session for photosynthesis? Okay. Photosynthesis just started, no? It's a brand new topic, no? Nothing is connected to all these. You can join this Thursday. I'm starting in the Thursday class, photosynthesis. Okay. Join and see. Okay. If I'm, okay. Let's see. Join and let me know what your feedback is. Okay. Hi. Let's move on. Polysaccharides. Come back to polysaccharides. Okay. Classification of polysaccharides. Okay, we are now going to classify. This is a complicated classification. Okay, so this space is for the classification of polysaccharides. Okay, listen. Classification of polysaccharides is the next topic that we have to do. Okay, you can classify, okay, polysaccharides. Okay, you can classify polysaccharides. Listen. based on two criteria. Based on two criteria, you can classify polysaccharide. First criteria, based on function. Based on function. Second one, based on architecture. What do you mean by architecture? Sort of the design of it, architecture. Based on architecture. Okay. Based on architecture. Based on function, that means what use do they perform? Okay. So based on function, you mainly have two functions of polysaccharides. They act as storage molecules. Then what is the other one? Then structural. Structural means they act as building. They act as building material. Then based on architecture, you can classify them in two ways. You can classify them as linear and branched. Linear and branched. Okay, linear and branched. So we'll come to this diagram. Can you see? This is, you can see someone is, this small child is holding some potatoes. If you zoom into the potato, you can have, in potato, what polysaccharide do you have? Starch. Starch is actually composed of two types of molecules, polysaccharide molecules, that are called amylose, amylopectin. Can you highlight it? Starch is composed of amylose and amylopectin. Can you see amylose are linear? There's no branching. But if you come to amylopectin, can you see it is branched here? Right? Amylopectin has branched. Right? Can you see? There's a branching here. So these two, amylose and amylopectin, get together to form starch. Now come to glycogen. Where is glycogen coming from? It's from this girl's or whatever child's hand. Okay, in your muscles, muscle tissue, you have a type of storage polysaccharide. Highlight this, glycogen, in muscle tissue. Glycogen is a very branched polysaccharide. Can you see there's a branching here, there's a branching here, right? There's a branching here. It's very, very branched. Glycogen is branched. Okay, so we can write down here under the based on architecture. I want you to write down, okay, examples. I want you to write down examples. So who is linear? Amylose. Amylose is linear. Branched, you can write down examples. The other guy, the friend of amylose, amylopectin, you can write as an example. And also glycogen, you can write as an example. Those are branched polysaccharides. Then you can add another one called hemicellulose. Hemicellulose, I'll explain. It is it's a friend of cellulose. Hemicellulose is a friend of cellulose. Then based on function, so you saw some, these are both storage. These are both storage. Amylose, amylopectin forming starch, and glycogen, they are both storage. So under examples for storage polysaccharides, you can write the combination of amylose and amylopectin. So these two both are forming starch. These two are forming starch. Storage, you can just say starch, and also glycogen, please add there. Glycogen. Don't write it. Mama, please wait a bit, otherwise you won't be able to manage your space. Okay. Please wait until I finish writing. Okay. Starch, glycogen. There's another storage molecule that I want you to know. Okay, that is called inulin. That is found in dahlia flower tubers. You can find this molecule called inulin. Okay, there is also storage. Then structural. Please come here. This green plant. Have you seen? We have zoomed into it. This green plant is composed of what? Plant cells. Can you see? There's a plant cell here. The plant cell has a cell wall. No? You learned this in grade 6 science, right? The cell wall is composed of these fibers that are called cellulose. Can you highlight? Cellulose. Cellulose is a nice linear one. Can you see how cellulose fibril, if you zoom into this, you can see how cellulose is formed. And if you take it out, it is composed of, Kian, what is this molecule, do you think? What is the building block of cellulose? What do you think? We'll come to these building blocks later. Okay, building blocks later. Can you see now what I want you to think about is cellulose is a linear molecule. Cellulose is linear as well as it is building, no? It is building the cell wall. So you can add a structural molecule example, sorry, a structural molecule polysaccharide example. You can write cellulose. And also, you can add another example here for linear molecules. You can add another example here for linear molecules. What is it? Okay. Cellulose. Okay? Cellulose is linear. You got null, strings. Looks like strings. Got it? Yes. Based on function, based on architecture, you can classify polysaccharides. Quickly take this down. In the page number 13, there's a space. Okay. If you want, you can take a screenshot. Okay. Because we need to cover a lot today. Anything you want to ask, clarify? Better if you can send to the main chat. Otherwise, I'm going to miss. Yeah, there's another one as pectin. I will add it. Okay, there's another structural pectin. Yes, yes, you can add it. Pectin. Pectin, I'll explain. Okay, what is pectin? I can show you. Okay, then anyone who's still writing? There's a nice uh, statement. Uh, my mom won't send me to your class because my friends are also coming. I will end up without listening. You can ask physical students who are coming if I give you time to talk at least with friends at my ISM or Headway class. I don't think students are chatting at all. Okay, because I try to keep their attention on me all the time. Okay, I don't think anyone is talking except in the interval. I give a bit of a long interval, 20 minutes. Otherwise, right? You can come, right? So, in physical students, I don't think they are chatting because I can see every one of you, and I don't think they are chatting. And I'm not going to sort of stop talking. I have never said stop talking in my classes. Okay, right. if the students are talking in your class, that means the class is not interesting enough. Ne? That means the class is not interesting enough. We'll come to functions of carbohydrates as a overall. Okay, we are going to start with monosaccharides, then disaccharides, and then we'll move on to polysaccharides. So I have given some diagrams here. We'll see all those. Okay. Monosaccharides is an energy source. Please highlight this. Monosaccharides are the commonest energy sources. Okay, so all the starch in the potatoes, rice, or bread you eat will be broken down into somehow into monosaccharides. Okay, will be broken down into monosaccharides. Got it? Okay, right. So monosaccharides main role, they act as energy sources, then building blocks of disaccharides and polysaccharides. So I told you they are like the bricks, right? Bricks that are building a big wall. Monosaccharides are the building blocks. Okay. Components of nucleotides. Can anyone tell me? So what are the components? What do you find in DNA? Which the monosaccharide do you find in DNA? Just told. Which monosaccharide do you find in DNA? Which monosaccharide do you find in DNA? No, deoxyribose. If you can't remember, you can write here. It is deoxyribose you find in DNA. In RNA, you find ribose. Okay. That's what I told you. Don't think about what you missed. Just stay in the current class. Then, otherwise, you are going to miss what you missed, and you are going to miss today. What's happening today as well. Just stay in the present. Forget about everything just for now. Okay. Then disaccharides, storage sugar in milk. Lactose is the sugar you find in milk. Please highlight this. Lactose is the storage sugar in milk. I don't want you to write this. You know these things. Phloem in plants. Okay. Highlight this word very nicely. Translocation is a very important word in phloem tissue. Translocation in phloem tissue is in the form of sucrose. Sucrose solution. Then storage sugar in sugar cane is sucrose, because that's how you in most parts of the world, that's how you extract sucrose from sugar cane. Okay. So that is disaccharides. Okay. Then here, this is a very important section. Here we are together while we are learning the functions, then we are learning the building block of the polysaccharides as well. So if you take, I want you to highlight this very nicely, because very commonly question. If you take starch, it stores glucose. Then what is the building block of starch? What is the building block or the monosaccharide of starch is what? What is the building block or monosaccharide of starch? Can glucose. Simple. That's because, can you see? If you go to this diagram here, all these are glucose molecules. All these are glucose molecules. You can draw an arrow. You can draw an arrow. These are all glucose. This is also glucose. Then this is also glucose. Glycogen is made up of glucose as well. Then glucose is the also the monomer in cellulose. This is also glucose. Glucose, starch, glycogen, cellulose. The monomer is glucose. Okay. Then highlight this next one. Glycogen stores glucose as energy in animals and fungi. Okay. Animals and fungus species have glycogen stores. Previous one also please highlight. Plants and chlorophytes. Can you write here what are chlorophytes? I wanted to write down here. They are green algae. Okay. Green, sorry. Because we need to write down their monomer here. Sorry. Chlorophytes are, listen, they are green algae. Green algae. You know, green algae that are floating on the seas. Have you seen some floating or ulva wage? green color sort of plant leaf like things that are floating on the ocean. Those are green algae. Then next one, inulin stores fructose highlight as energy storage in Dahlia tubers. Okay, this inulin powder contains fructose. Some people use it as a dietary supplement. Inulin powder, they add it to their food. Okay, you can Google and see later. Right, let's write down the monomer. Let's write down the monomer. Okay, so if you are too lazy, you can put a mehemekak. Okay, then you can say glucose. Then in inulin, what is the monomer? Fructose. Okay, write with me as as I soon as I tell you to write, you have to write. Okay? Right. So we'll have a small discussion after carbohydrates are done. Okay. Then we move on to the next function, that is structural polysaccharides. Structural polysaccharides, that means they build things. Cellulose in the cell walls of plants and chlorophytes. Please highlight. In your plants and green algae, you have cellulose. Pectin in the middle lamella. Please highlight this. And I want you to put a red underline as well if you can. Middle lamella is a very important word. I'll explain what is the middle lamella. Okay. Next, hemicellulose, highlight. Cell walls of plants. Hemicellulose is like a friend of the cellulose. They are therefore sort of the give some additional support to the cellulose fibers. Okay. Give some additional support to the cellulose fibers. Okay. Peptidoglycan, this is not entirely essential because we'll learn about this later, bit later, but I added it here because it's nice to know. Cell walls of prokaryotes. Can someone tell me what are the prokaryotes you know? Some examples. Bacteria are the main prokaryotes. Right? Yes. Example, write down bacteria. Chitin, finally, chitin in the cell walls of fungi, exoskeleton of arthropod. Cell walls of fungi. Fungal cells have a cell wall. Fungal cells have a cell wall. Exoskeleton of arthropods. Exoskeleton of arthropods. What do you mean by an exoskeleton? What do you mean by an exoskeleton? What do you mean by an exoskeleton? A skeleton that you find outside the body. Meti. Can you see this whatever some sort of bug is so who is the bug here? You can see two bugs again. Bug or insect. Who is the living one? Man, this is the living bug. Okay. So what is he leaving behind? His old skeleton. So have you ever gone to your kitchen at night, kitchen or bathroom, washroom at night and accidentally stepped on a dead cockroach accidentally? It's very crunchy, no? Crispy. Okay, that is because that is outer shell is made up of chitin. This is the exoskeleton. That is a skeleton on the outside. Okay, that is composed of chitin. So you can write here. You can write below this diagram, right? You can write below this diagram small sentence. Okay, you can write below this diagram. Molting means sort of you are removing the old exoskeleton. Molting of the exoskeleton is shown here. Molting, that means, for example, imagine you are living inside your skeleton. That means when you get bigger, your skeleton is going to restrict you, right? So as you are becoming a big insect, loku insect you have to remove your old skeleton and build a new one. So this insect is doing that. That is called molting. molting, molting. Can anyone tell me an animal, not an insect, that is having an exoskeleton, a skeleton on the outside. Some general knowledge, an animal. A snake skeleton, haven't you seen? A snake has a very long, sort of a backbone like skeleton, right? A snake has a very long mehima from the head. There's a skull and it has inside. The skeleton is inside. You are thinking of this snake's scales. Scales are just there for protection. No, no. Someone who has a protective cover outside. They are living inside. No, okay, now you have to think. I think. Yes, turtle or tortoise. Turtle or tortoise. Crab is also a crab. Yeah, what do you think? Crab is a crustacean. Crustaceans are also arthropods. Okay. So, yes, turtles and tortoises, they have a nice shell. Okay. Right. So, I want you to write down the monomers. So, under the list of monomers, for cellulose, who's the monomer? Glucose. Yeah. Cellulose, what is the monomer? Glucose. Then what else do you have? Pectin. Pectin is, the monomer is called galacturonic acid. Galacturonic acid. Okay. Hemicellulose is actually composed of a variety of molecules. Hemicellulose is not a fixed molecule, it's actually a family of molecules. Okay, so we say it could be composed of a variety of pentoses. That means you can have 5 carbon, pentoses and hexoses. Okay, it can be composed of a variety. Okay, right. Then peptidoglycan. Okay, you don't need to know. Peptidoglycan, you don't need to know, so you can keep it blank. Okay, you don't need to know the monosaccharide. Then chitin, yes, chitin, the monosaccharide is called glucosamine. Glucosamine. Why is chitin classified as a carbohydrate even though it contains nitrogen? Yes. What is the main requirement for something to be considered as carbohydrate? It should contain the same hydrogen to oxygen ratio as water. Hydrated carbons. So even though it contains nitrogen, okay, you can't say it's not a carbohydrate because it contains the same water H2O ratio. Yes, so that's a good point. Methana, I want you to put a star mark here and put an arrow to chitin contains nitrogen. Contains nitrogen. Contains nitrogen. Okay, so can you see everything is organized like this? So the monomers and the monomers of the each of the polysaccharides you need to be able to remember. Dahlia tuber is like a root or a alec, right? Something that is grows underground in dahlia plants. It grows underground. So inulin is something that is like it's a storage form of storing energy. Like in potato you have starch, in dahlia you have inulin. It's powdery form. Got it? Inulin. Inulin, okay? So carbohydrates are done. Then I'll explain quickly what is this middle lamella. We'll talk about the middle lamella later. We'll add a middle lamella here. There's some space now. We'll draw a small middle lamella here. Taggalla, you need you to draw with me, okay? Otherwise, we won't be able to cover. So, if you take plant cells, listen. Plant cells are sort of polygonal. Plant cells have a plant cell wall. Plant cells have a cell wall. Plant cells have a cell wall like this. Take another plant cell that is the neighbor of this plant cell. The neighbor of this plant cell is this. Then there's another plant cell here because that's how a plant tissue is formed. Okay. So imagine these plant cells are again like bricks of a wall. Bricks of a wall. Somehow, if you build a wall out of bricks and you don't paste them together, just bricks stacked on top of each other, that's not going to be strong, right? If you push the wall, it's going to fall down. Yeah. So somehow you need to paste these plant cells together when you're making a plant tissue. So the cement, so like for the bricks, in between bricks you have cement. In between your plant cells, you have a cement that is called as middle lamella. Middle lamella. A cement which is called as middle lamella. Okay, so quickly draw some plant cells. Okay, I want you to label this is the cell wall. Then the cement is called the middle lamella. Then you can the inside the plant cell you have the cytoplasm, right? Normal cytoplasm, cell wall. This is the middle lamella. Okay. Okay. Any connection issues, people? I see people coming and going. Any issues? So are you going and eating something and coming back? What's happening? Okay. Cell wall. Plants are bound by a cell wall, and in between those cell walls, you have a cement that we call as inulin. I'm sorry, the middle lamella. Okay, middle lamella. Okay. Okay. So carbohydrates are over, right? Simply put, middle lamella is actually it's a mineralized. Okay, it's actually composed of calcium and magnesium ions are also there, but the main composition, remember, it is pectin. Okay, main composition of the middle lamella is pectin, because if you take like it's the cement between plants. Now, how do you make cement? You need cement means a mortar, right? That's you don't just put cement. What do you need to make the mortar? Badam killing. You need sand, then cement, water. Similarly, when you are making this middle lamella, the main thing is pectin. Then you need sometimes calcium, magnesium to make like a nice badam, a nice mortar. Okay. No need to know the monomer of peptidoglycans. Just remember found in cell wall of prokaryotes such as bacteria. Okay. Okay. So carbohydrates are over. Let's do an MCQ quickly. Let's do an MCQ quickly. I want all of you to send answers to the chat. Okay. This question, quickly do this. Which of the following is a non-reducing sugar? Which of the following is a non-reducing sugar? Should quickly send me the answer. Okay. Others? Yeah. What is the only non-reducing sugar? Sugar means what? It is has to be either a monosaccharide, okay, disaccharide, monosaccharide or disaccharide. Then what is the only uh, non-reducing sugar that you have? Sucrose. Okay. Then I want you to listen. Whenever we are doing MCQs, you might have seen in our normal theory class, we like to write reviews, right? Whenever in front of the question, we like to write reviews because from the question you learn theory. That is the most effective way. Got it? So in front of this, I want you to write down a small review. Sugars are of two groups: monosaccharides and disaccharides. Write down a small sentence. Sugars are of two groups as monosaccharides and disaccharides. Sugars are of two groups as monosaccharides and disaccharides. Okay, right. This one, okay, we skipped some, no? Right. You can't do these things. Proteins. Yeah, can you quickly do this one? What is the monomer of each of the following? These are all past paper questions. This is in the same tute. Okay, same tute, last pages, page number 22. Okay, 22, 23. What are the monomer of each of the following? Quickly write, quickly write. Okay. Pectin, galacturonic acid, yes. Hemicellulose, it could be a variety, no? So it could be pentoses and hexoses. Mix and match. Okay. Pentoses and hexoses. Okay. We'll do one more and we'll move on to quickly lipids. Okay. Quickly do this one also, right? Quickly do this one also. Correct regarding carbohydrates. Send me the answers, okay? Okay. So I hope, this is the best way to not to fall asleep, huh? Instead of going along doing theory. Okay. Just do a question and send me the answer so we can discuss it together. So you get the time to think. Okay. How to memorize stuff quickly? The best way I have found is by visualization through diagrams. Okay, that's why I try to include a lot of diagrams. So now you can remember what a exoskeleton is, because I have shown you an exoskeleton. So since you have seen an exoskeleton, then the next step is to remember, okay, exoskeleton is composed of chitin. Okay, right. Following statement is correct regarding carbohydrates. Condensation reaction between two glucose molecules from a non-reducing sugar molecule. What do you think? Two glucose get together to form a glue plus glue flows. क्या ना maltose नहीं तो maltose is a reducing sugar. Please write this. Glucose plus glucose produce maltose. Maltose is a reducing sugar. इतनी लिया गया ना maltose is a reducing sugar. So it's false. नहीं write down there. Then galactose is a ketose sugar. No, it's an aldose. Neither. Who is the only ketose sugar that you need to know? You can draw an arrow. Fructose. Neither. Fructose. Galactose is a aldose sugar. Then glucosamine is the monomer of storage components in fungi. Right? Listen very carefully now. This is a tough MCQ. What is the storage component in fungi? Mulima, first identify that. What is the storage component in fungi? Yes, that is glycogen, no? Storage. What is the storage? Something stored in fungi is glycogen. Then, what is glycogen composed of? Glucose. So, this is false again. You can draw an arrow. In fungi, it is glycogen. Glycogen is composed of glucose. So, you can remember, yes. Make your own reviews, right? For particularly for physics, you will understand for past paper questions. I think by now you must have bought a Pesur book and started trying some past paper MCQs. The best skill you need to develop for physics MCQs is to write reviews. That means how to do this question in your own words. the equation I apply or the logic behind this in your own words. Okay, but I try to give you a sort of a model review so that you can learn something from the question as well. Galacturonic acid is the monomer of structural polysaccharide in plants. What is the structural polysaccharide in plant? Yeah, so you can put an arrow, that is pectin. No? Pectin, then you can draw another arrow, found in middle lamella. This is a 2024 level question. Okay, found in middle lamella. Hemicellulose is a branch polysaccharide made up of trioses and pentoses. Made up of trioses and pentoses. Hemicellulose? No, you can correct this. Cut it with a red pen. We do this and something called correcting the MCQ. So sometimes that's the only review you need. Okay, pentoses and hexoses. You correct it. Just correct it. Okay, we'll move on to lipids. Okay, so before we move on to lipids, let's take a small break. Before the break, I do you want to see? This is something I recently saw because I'm doing actually not this particular appointment. I'm in the Apeksha hospital doing oncology, cancer related medicine. Okay, so this is something we do called endoscopy. Okay, this is something we do called endoscopy. That means we put a camera. It could be from your mouth, it could be from the bottom. Okay, so endoscopy. So if you are interested in what your vocal cords looks like, this is how your vocal cords look like uh, from this camera view. Okay, so this person is sort of making some sounds because it's very sort of discomforting. Can you see these two are your vocal cords? When these two are the vocal cords. This person is, so the saliva is there, right? So when you are speaking, right? Can you see the vocal cords are going to get bigger and tense. When they are usually close together, they make high pitch sounds. They make high pitch sounds. When they are sort of wide open, you make sort of the bass sounds. Got it? Can anyone tell me what is this? This front structure, this one. What is this? Can anyone tell me what that is? This sort of leaf-like thing, this front one. You learned about this in O levels, related to something called during swallowing, when you swallow food. Okay, so the camera gets covered with the person's saliva, so sometimes the view goes away. Okay, so we couldn't go past. We should go past, not to the epiglottis. Actually, sort of here in front, you have the esophagus. We are supposed to go to the esophagus, but that person was not very cooperative because they started to cough and started to feel discomforted. So we had to stop. That is not my hand. Okay, right. So that is called epiglottis. I'll show you. This epiglottis helps you to not have any food in your lungs. Not to go in the wrong pathway. Epiglottis. Okay. I think I have a small video in my YouTube channel also. I have a small video. I can explain to you swallowing right now. Swallowing. Oh, maybe. Right.
Digestion begins in the oral cavity, where the salivary glands, teeth, and the tongue work to break food
Can you
down
hear the
into
audio?
small masses that can be swallowed. The movement of the jaw enables the teeth to grind food. Saliva secreted by salivary glands aids the mechanical and chemical process of digestion. The tongue manipulates the chewed food into a small mass, then moves it into the oropharynx. The next steps are involuntary. The bolus passes.
Can you see? So this is the epiglottis. So there are two pathways. This is the food tube. This front one is your windpipe. If food goes into your windpipe, you can choke. Okay, your food contains bacteria also. You can get a lung infection. So your epiglottis is going to stop food
Through the
going into the
pharynx,
wrong
the
pathway.
epiglottis closes off the trachea, and peristaltic waves move the bolus into the stomach.
Got it? So I just wanted to show you that. Okay, that's enough. Take a small break. 5 minutes again. Then we have a lot to cover today. Take a small break. Got it? Quickly join. 10 minutes is enough, right? 10 minutes is enough. Okay. When did I give the break? 8:01. Shall we continue? Go ahead. Yeah. We can take the introduction into lipids. Lipids is a short topic, okay? Lipids is a short topic, so we'll see how much further we can go within today. Okay, let's try to cover maximum. Because my goal is to bring you up to the level of the normal theory class as soon as possible. Then from your mind, you'd have that, you delete that idea that, okay, I started late, I started my after my O-level results, which is actually the right way to do it. Because our tuition culture has generated, okay, you have to join as soon as you finish your levels, but you are now qualified. You got whatever result, and now you are qualified. So this is actually the right time to start this. Okay. Okay. Did you Excel low level? Can you do a local level? Yes, of course. Can you do means what? Are you would be able to physically do it, or I mean, is there something preventing you? No, it's actually good because from your Excel knowledge, you have your English is already good. No, if your English is already good, I mean, then you don't need to worry about catching up. But I mean, I recommend that you pay a bit more attention to physics and chemistry because in physics and chemistry you need to apply logarithms. You can't use calculators. There are some things you need to consider. No? So those skills you can build up slowly. Yeah. Okay, lipids. Diverse group of hydrophobic molecules. Diverse group of hydrophobic molecules. Can you highlight the word hydrophobic? Hydrophobic. That means phobia means what? You are afraid of something. Right? So lipids are kind of afraid of water. Hydrophobic means you can say it doesn't dissolve in water or doesn't mix with water. Hydrophobic. Okay. Large biological molecules, but not considered as polymers or macromolecules. Okay. Very, very important. Please highlight not considered as macromolecules or biopolymers. Lipids exist as big separate molecules. Big separate molecules. They don't form chains or polymers if you like you find in polysaccharides. Right? So you have big molecules, but they don't connect with each other to form long chains. Okay, so that's why they say they are large molecules, but they are not considered. Okay, next. Consists of carbon, hydrogen, oxygen, but the hydrogen to oxygen ratio is not 2:1. Please underline the word not 2:1. You have relatively more hydrogen. That is very important. You have relatively more hydrogen. You have relatively more hydrogen. Therefore, that's one of the main differences of lipids. Biologically important types of lipids, we have actually three types of lipids that we are going to talk, but you need to remember the three. but we are going to discuss only these two, fats and phospholipids only we are going to discuss. Fats and phospholipids only we are going to discuss. Steroids, we don't learn about the structure, we learn about some of the examples for steroids only. Okay. Where are the other people still having the break? Between large molecules and macromolecules actually they have, I can't remember the exact value, there's a molecular weight limit. I mean, above that molecular weight you consider as macromolecules, large. I mean, they are sort of above that molecular weight value, they are considered as macromolecules. So you will see when we learn about lipids are sort of complicated molecules, as we'll draw later, right? Lipids are complicated molecules like this, but they are not big enough to be considered as macromolecules. Okay, they are not big enough to be considered. But the main difference is it should act like a polymer, right? It should have a distinct monomeric units to be considered as a macromolecule, and a biopolymer to be considered. You should have building units, right? For example, in a polysaccharide, you have monosaccharides. It's like that. Okay, let's talk about fats. Very common. Fats are made up of glycerol and fatty acids. Those are the two components. Glycerol belongs to alcohol group having 3 carbon atoms, where each of them bear a single hydroxyl group. Okay, so glycerol is an alcohol with 3 carbon atoms. Okay, right. So let's draw an glycerol here. Let's draw a glycerol here. Glycerol has 3 carbon atoms. 1, 2, 3 carbon atoms. Okay, glycerol has 3 carbon atoms. Can you hear me? Yeah, any audio issues? No, right? Then as an alcohol, you will learn in organic chemistry, alcohol group bona alcohol, the drinking alcohol is only one of the alcohol family. It is ethanol, right? Alcohols, we say they contain OH groups connected to a carbon. So this OH groups makes it an alcohol. This is glycerol. This is glycerol. 3 carbons with 3 OH groups. 3 carbons with 3 OH groups. Okay. Right. Then what is the other part of the fat? Fatty acids. Fatty acids are hydrocarbon chains with long 6 to 18, highlight the number, carbon skeleton with a carboxyl group at its one terminal. Right. What is a carboxyl group? What is a carboxyl group? That is also a type of a family of organic molecules. It's a family of organic molecules. Listen very carefully. A carboxyl group has a carbon double bond oxygen. Okay, carbon double bond oxygen, and bonded to that you have a OH group as well. You have an OH group as well. How many fatty acids do you need? How many fatty acids do you need? You need 3 fatty acids to form one fat molecule. You need 3 fatty acids. So I'll draw another fatty acid. This is the carboxyl group. Pay attention. Okay, this is the carboxyl group. This is the other carboxyl group, OH. Then what else do you have? Fatty acids have a hydrocarbon chain with a carboxyl group. So what do you mean by a hydrocarbon chain? Hydrocarbons. That means hydrogen containing carbon chain. So I can draw carbons, 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 carbons. How many? Could be between 16 to 18, but actually in reality it could be 21, 22, 20. So fatty acids, there's a large number of fatty acids. But just remember in your syllabus, they say 16 to 18. So here also, you can have carbon, 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 carbon. Okay. Then what makes it a hydrocarbon chain is that you have a hydrogen, 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 hydrogen. This is a hydrocarbon chain. But in organic chemistry, we don't draw this carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. That is for chuti lamai, right? Small kids. people who are just learning organic chemistry. So in bio as well as in organic chemistry, if you have a very long hydrocarbon chain, you can indicate that hydrocarbon chain in a sort of a shorthand. You can draw it in an easy way. You can draw it in an easy way. That is, you can indicate one carbon hydrogen with a sort of this point of a line. This point contains a CH, and this point contains another C double H. So you can draw this hydrocarbon chain with a zigzag line. You can draw it with a zigzag line. Turn, 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 turn. You can draw it with a zigzag line. You can have zigzags up to 16 to 18 zigzags. Okay. So here also you can have up to 16 to 18 zigzags. Here also you can have zigzags. So what is this molecule containing of a carboxyl group at the one end and a long hydrocarbon chain, that is a fatty acid. Okay, that is a fatty acid. Are the people not sleeping either? Yeah. I usually say online people not sleeping either because now it's only online people. So okay. In response, the chat is very boring. What's happening? Earlier there were some questions. What sort of a doctor do I want to be? I just want to be a. What sort of doctor do I want to be? Okay, right. Let's talk about that later. So this is the recipe. I'll shorten this. Okay, all my fatty acids went away. I'll shorten this. Otherwise, I won't have space to draw the output. Okay. So this is how the fatty acid molecule looks like. What is the composition of the fatty acid? It has a carboxyl group and a hydrocarbon chain. Okay, this is how you indicate the hydrocarbon chains. Then now, so you have, this is the recipe. This is, you combine these two to form a fat molecule. Okay, you combine these two to form a fat molecule. Highlight, okay. Fatty acid molecules bind to hydroxyl group of glycerol by, okay, now you are learning the second type of bond in this organic molecules. Highlight this ester bond. Ester bond. Then this bond formation. So there's going to be a bond formation. We actually call it esterification. Esterification process happens. Then you are going to get what? A fat molecule. So you have a glycerol. You have a glycerol with what? I'll indicate the hydrogens like this. Okay, CH and CH2. Okay. Now, what sort of a bond is this ester bond? Ester bond, listen carefully, it is also a dehydration reaction. Ester bond is also a dehydration reaction. The hydrogen will be given by the alcohol of the glycerol, OH by the carboxyl group, the blue color group of the fatty acid. This is very, very important. You need to remember who gives the hydrogen, who gives the OH group. Okay. The hydrogen is given by the OH of the glycerol. The OH is given by the the carboxyl group of the fatty acid. Okay. So now what you have? You have this O group. You have a nice new bond that is called as an ester bond between this oxygen, this line of oxygens, and this carboxyl carbons. Okay. So you have carbon, carbon, carbon. Carbon has a double bond oxygen, double bond oxygen. Then you have your hydrocarbon chain. This is the output. You can label this bond. What is this bond? Yes, ester bond. Anything I need to add to this reaction? Anything I forgot? To make this a balanced reaction, what did I forget? What did I forget? I told you it's a dehydration reaction. Yes. I need to add a Water molecule. How many water molecules? How many water molecules? 3. Yes. 3H2O, if I add to this side, the equation is balanced. Okay, quickly take this down in the space. Okay, quickly take this down. Draw the glycerol molecule and 3 fatty acids. Okay, then esterification is a type of dehydration reaction. You're going to end up with a, what is this? Then I'll hold on. Then we'll start drawing. Okay, this is your glycerol molecule plus 3 fatty acids. This is the recipe. I have run out of space. 3 fatty acids is going to end up with a, okay, fat molecule, or you can call it, listen carefully, triacyl glycerol. Triacyl glycerol molecule plus 3H2O. This is the equation or the recipe to form fat. The recipe to form a fat. So you get different types of fats based on the different types of fatty acids. You can have a large variety of fatty acids. Could be plant fatty acids, animal-based fatty acids. Sometimes you artificially make fatty acids in these food, in factories, in labs, right? So you have all your different types of fats. You got also different types of fatty acids. Okay, write this. This process is called esterification. You are going to end up with a triacyl glycerol. The word is given here as well. Triacyl glycerol. Okay. This is also a condensation reaction. Yes, a condensation or a dehydration reaction. Okay, this is also a condensation or dehydration reaction. If you feel like I'm not giving you enough time, you are free to take a screenshot. Okay, because there are, I need to cover some topics. Can we genetically inherit thyroid cancers? Cancers are not mostly genetically inherited. They are acquired because of certain things in the environment. Could be radiation, could be diet. Okay. Some amount of genetic influence can be there, but you can't inherit the cancer. There can be some factors that can make you likely to develop an abnormal cell. That abnormal cell become a cancer. You are not born with cancers. Okay. Right. Okay. Then keep reading. Very important statement. The hydrocarbon chains contribute to the hydrophobic nature. Your hydrocarbon chains, that means these people, they are hydrophobic. Understood? They make the fats afraid of water. If you take some butter, can you dissolve it in water? No. That is because of their hydrocarbon chains. Based on the nature of the hydrocarbon chains of the fatty acid, fats are of two types. Can you understand? What is the nature of based on the nature of the fatty acid, fats are of two types. So, if a fat is composed of fatty acids without any double bonds, no double bonds, Haridha, listen, that fatty acid is called a saturated fatty acid. Then the fat that is composed of saturated fatty acids is called as, any guess? Saturated fat. Right? What do you mean by saturated or not having any double bonds? What do you mean by not having any double bonds means, can you see all the carbons are connected by single bonds. So if you can just draw a simple hydrocarbon chain, mehema, all the carbons are just connected by single bonds, single covalent bonds, you know the bonding, single covalent bonds. So why do we call them saturated? Because you have put the maximum number of hydrogens possible. You can't put any more hydrogens into this hydrocarbon, no? So highlight some examples for saturated fats such as butter. Butter is a saturated fat. They are mostly solid at room temperature. So you might say, okay, butter melts in room temperature. When you put them outside on the fridge, they melt. No. Why does that happen? That is because our room temperature is much higher than standard room temperature. The standard room temperature is around 25 degrees Celsius. Our room temperature is how much? Around 32, 31. Okay. So butter is going to melt. A good quality butter at around 25, 26 degrees Celsius is not going to melt. That is because it is composed of saturated fats. So highlight, usually animal fats are saturated. Then the next classification, listen and highlight. If a certain fatty acids hydrocarbon chain have one or more double bonds, they are called as unsaturated fatty acids. And that fat that is made of unsaturated fatty acids is known as unsaturated fats. Okay, highlight, usually plant fats are unsaturated fats. They are mostly liquid, such as your vegetable oils. Highlight the examples. Plant fats are usually liquid at room temperature. Plant fats are usually liquid at room temperature. Okay. So we have some clinical scenario to discuss, how you get heart attacks and other things. How do you get heart attacks? We have to talk about. Okay. Okay. Then unsaturated fats, you can classify them based on the nature of their double bond. This is something again a bit chemistry. Okay. There are two types as cis unsaturated fat and trans unsaturated fat. Okay, listen. A cis unsaturated fat that is given here, in a cis unsaturated fat. So you know, if it's unsaturated fatty acid, there should be a double bond. No, if there is a double bond, that means the carbon can bind with only one more hydrogen, because why? Carbon can only form 4 covalent bonds. So here already you have 1, 2, and 3. So it can bond with one more hydrogen. This carbon can also bond with another hydrogen. This, if you have both hydrogens on the same side, that is called as cis. If you have both hydrogens on the same side, that is called as cis. Can you draw this? This is cis. Then, if you have if you have hydrogens in the double bonded carbon on the opposite sides, that is called trans. Trans means sort of opposite, right? The higher carbons are located on the opposite ends. Quickly draw the cis and trans. You will learn this obviously in chemistry later. Okay. This is cis. If you have on the same side cis, opposite sides the hydrogen trans. Quickly draw this. So cis and trans are the two types of unsaturated fatty acids. So when a fat is composed of those unsaturated fatty acids, you call them a cis fat or a trans fat. They are both unsaturated. So highlight, they are both unsaturated. Okay, then there's a very important statement here. If you can highlight it in or you can underline it in red color, excess saturated fats, okay, consumption of excess saturated fats, okay, highlight, excess saturated fats and trans unsaturated fats contribute to atherosclerosis. Underline this word, atherosclerosis. That's what I'm going to explain next. Atherosclerosis. Atherosclerosis. So, if you usually consume a lot of animal fats, particularly in sort of fatty meat like pork, or I mean, good healthy butter have a lot of additional benefits, but if you consume a lot of butter, particularly animal fats, pork and chicken can have some fats based on the cut, chicken legs and those, where else? Yeah, chicken skin can have fat. If you consume a lot of those fat, saturated fats are also found in the egg yolk as well, but they have a lot of benefits. Can you see the word here? Excess excess saturated fats. You need saturated fats in your body, in your diet, but if you consume more than you need to, there's going to be problem. ඕනම දෙයකට ඕනට වඩා ඕන එකක් මොකද එහෙම ඇති ඕනම දෙයක ඕනට වඩා ඕනෙම නැහැ මොකක් හරි. You don't need excess of anything. Okay, saturated fats are good, but when you have excess, there could be problem. But listen, trans unsaturated fats, any amount could be harmful. Any amount of trans unsaturated fats, dietitians say do not eat trans unsaturated fat. Trans unsaturated fats are usually artificial. Okay, give me an example. Your fat spreads. Fat spreads. They are not butter. Okay, they are not butter. Usually, trans unsaturated fats are found in fat spreads. There are so many brand names, fat spreads. Okay. Earlier, they were all marketed as margarine. Now, they have fat spreads. Those are very, very artificial. They are composed of certain vegetable oils, palm oils that they have chemically altered. Okay. There's a question, what about ghee? Ghee is an animal, it's extracted from milk, no? So, it contains milk fat. Milk contains saturated fats. Yes. Ghee contains saturated fats. Ghee. Okay. It's very tasty. It could be nutritious, but only in enough quantities. Okay. let's come to this page. I want you to write down a small topic here, atherosclerosis. Okay, atherosclerosis. I just finished this to my 27 batch because we are doing the blood circulatory system and in the heart. We talked about the problems that you can face when you have atherosclerosis. Okay, atherosclerosis is simply this. What has happened to this blood vessel? This is a blood vessel. Blood vessel due to some fatty deposits. Fatty deposits. Can you see the blood vessel wall has become narrowed? So the blood is not able to flow nicely. Okay. If this happens in some part of the heart muscle will not get enough nutrients and oxygen. Heart muscle will die. You end up with a dead heart muscle that we call as a myocardial infarction. Myocardium is the heart muscle. Infarct is a dead area or a heart attack. Okay. Right. So let's write down the definition of atherosclerosis. This is the definition that you have in the syllabus. Okay. This is not a nice, say, ideal one, but not a medically accurate, but it's the one you have in your syllabus. Okay. So write down here, thickening and hardening. Write down with me, thickening and hardening of inner lining of arteries due to fatty deposits such as cholesterol is atherosclerosis. Okay. Thickening and hardening of inner lining of arteries due to fatty deposits such as cholesterol. Okay. Atherosclerosis. Then one more point, you can write it in a star mark. Blockage of cardiac, sorry, blockage of, I'll explain. Okay, let's write down with me. I don't have space. Your handwriting should be smaller than mine. I'm writing on the smart board. Blockage of coronary arteries due to atherosclerosis can cause myocardial infarction. Write down. Blockage of coronary arteries due to atherosclerosis can cause myocardial infarction. Can cause myocardial infarction. Okay. What do you mean by coronary arteries? Anyone? What do you mean by coronary arteries? The arteries supplying blood to the heart. I'll quickly show you. Mega, have you written this? Am I going too fast? Anyone? You can let me know. Am I going too fast? Yeah. Going too fast? Anyone? No. Others? Any comments? Difficult to understand? So I'll show you the blood supply of the heart quickly, okay? If you take your heart, okay, if you take your heart, you can see it's an open view. Now, these are the ventricles, right? You know the ventricles. These are right ventricle, this is the left ventricle. So, the heart is composed of cardiac muscle, or heart muscle. It's very, very metabolically active. Okay. So, the heart muscle requires a lot of oxygen to keep beating and also a lot of nutrients for energy. Okay. And you need to remove waste. So, the heart itself has a blood supply, no? The heart pumps blood, yes, but someone should give blood to the heart also. That is done by a group of blood vessels called coronary blood vessels. Out of them, the blood vessels that bring blood to the heart are called coronary arteries. Those are the red color ones. Can you see? Can anyone name this big blood vessel that is going out of the heart? Big one that is curving like this? The aorta. From the aorta, can you see you have two blood vessels coming out? There's one here, and if you look at here, there's one here. Those are the main coronary arteries. If those coronary arteries, can you see they are going? So this front one is going down like this and wrapping around the heart, and this back one is sort of branching like this and this, right? If this atherosclerosis or the narrowing of the blood vessels occurs in one of these main arteries, the heart muscle will not get enough oxygen or remove its waste matter, so that the cardiac muscle will die. So the heart muscle is heart having some dead areas means the heart is not a very good pump. Then your body will not have enough oxygen, and eventually you will die. You will body will not have enough blood pumping. Okay, that happens due to atherosclerosis. There are other reasons for having heart attacks, but atherosclerosis is one of the most common. Okay. Okay, come back to the tute. Then we'll talk about the next type of lipid that we have to talk about, that is called phospholipids. Phospholipids. Okay, phospholipids. Let's read. Phospholipids are major components of cell membrane. So very soon we'll be discussing the plasma membrane. Phospholipids are major components of the cell membranes. Okay, what are the parts? So this is the recipe to make a phospholipid: two fatty acids and one phosphate group attached to one glycerol. Those are the main things. Can you see? You have two fatty acids. Can you see here? You have two fatty acids. This is your glycerol. This is your phosphate. Additionally, depending on the type of phospholipid, you can have additional polar molecules such as choline. Okay, can you see choline here? Okay. Then very important statement is there here. How do you highlight? Phosphate groups gives a negative charge to the phospholipid. So always remember, phospholipids are negatively charged molecules. Phospholipids are negatively charged. Can you see here? This phosphate has a negative charge. Yeah, PO4 3 minus, but we have neutralized the other oxygens. Therefore, you can say the phosphate group has a negative charge. Okay, this is our sort of the more structural formula of the phospholipid. This is the 3D model or the space filling model. Okay, then there's a symbol. That is the one you need to know as a bio student. That is the one you need to know as a bio student. How to symbolically draw a phospholipid. How to symbolically draw? That is, you draw a ball like this. That is called the head. Okay. Then you draw two tails like this. Give me a name for those. They are tails. Okay. A phospholipid you draw with a head and two tails. Okay. So then there's an important statement. We are going to read that. Two ends of the phospholipid show different behavior. Okay. The hydrocarbon tails are hydrophobic. Please highlight that. Hydrocarbon tails are hydrophobic. Phosphate group and its attachment or the head is hydrophilic. Can you see the differentiation? The hydrocarbon chains of the fatty acid. Now, I hope you can remember what a fatty acid is. A fatty acid is a carboxyl group attached to a hydrocarbon chain. So, how is hydrophobic? Only the hydrocarbon chain of the fatty acids. Okay, the entire fatty acid is not hydrophobic. Only hydrocarbon tails of the fatty acids. Okay, who is remaining then? Who is remaining of the fatty acid? Who is remaining? If I magnify it, who is remaining from the fatty acid? This, the carboxyl groups of the fatty acid is remaining. Yeah. So only the hydrocarbon chains of the fatty acids are hydrophobic. So who else is hydrophilic? Who else likes water? This whole thing, no? Can you see? The hydrophilic head. Hydrophilic means the opposite of hydrophobic. That means you like water. You are a fan of water. You have a sort of a relationship with water. You like water. Got it? So the hydrophilic head. All this is the hydrophilic head, right? All these things contribute to form the hydrophilic head. Hydrophobic tails are only, I want you to write down here. What are the things that are included in the hydrophobic tails? Can hydrocarbon chains of fatty acids. They form the hydrophobic tails. Then what do you find in the hydrophilic head? You have the phosphate, glycerol, and carboxyl groups of fatty acids. FA කියලා කියන්නේ fatty acids. Those are the things that are included in the head. Okay. Not sleepy near the kattiya. Okay. Right. Draw this phospholipid symbol. Do not go into this space. Save some space here. Okay. Can you quickly draw this? This is the symbol of a phospholipid. This is how you draw a phospholipid. It has a head and a tail. So could you label the head also? This is called hydrophilic. It likes what? Hydrophilic head. These are hydrophobic tails. Quickly draw this. Okay, right. Phospholipid, that is how you draw phospholipids. Then let's come to this, what did we say? Two ends of the phospholipids show different behavior. They can head dekai tail dekai. In the same animal, the head and tail behaves in two opposite ways. Okay, right? So, for example, in your home, your father could be supporting one political party, your mother is supporting one political party. So, they are their minds are two different philosophies. Like it's like that. So, if you take an animal cell, usually in your body cells also, we live in a water filled environment. So we know that water is the biological medium, right? So inside cells as well as outside cells you have water. So when we say our cell membranes, cellular membranes are composed of phospholipids, but what's the problem? Your hydrocarbon tails of the fatty phospholipids don't like water. So somehow they should avoid the water. Only the heads should face water. So they found a solution. The phospholipids found a solution. Let's make the heads face the water inside and outside. Let's make what? Let's make the heads face the water inside and outside cells. So this is the, let's say, inside of the cell, you have the cytoplasm. No? In the cytoplasm, you have water. This is the outside of the cell. No? Imagine it is one of your body cells. Outside your body cells, you have blood. Yeah. So that is also mainly water. No? So you can say this is intracellular, that means inside of the cell, intracellular, right? Be familiar with these words. So you will use this naturally. Okay, inside of the cell is intracellular, outside of the cell is kya extracellular, right? With time you will learn these phrases very easily, right? Then so these heads are protecting the tails are like in the inside, like this. The tails are facing each other so that they are protected from the water, because they don't like water. You can even say they are afraid of water. Hydrophobic, no? That is their chemical nature. So don't ask me why don't they like water. That is how hydrocarbon chains behave, because hydrocarbon chains can't form bonds with water. Hydrocarbon chains can't form bonds with water, therefore they don't like to exist with each other. Okay. So then what I want you to do is, okay, so can you see this is called phospholipid bilayer. In front of, on top of that, there's a blank space. No, can you draw this? I want you to have intracellular, extracellular. This is called the phospholipid bilayer. Bi means what? Bi means two. Okay. 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 Right. So take a screenshot. Now you can see how properly complicated the actual phospholipid bilayer forming the cell membrane. This is called the plasma membrane or the cell membrane. So these are all the hydrophobic tails, you can see these are all the heads. These are the heads. Okay, understood? This is how the complicated cell membrane looks like. We'll talk about the cell membrane probably next week. Okay, in the up to speed sessions. So this is how this phospholipid bilayer forms the plasma membrane. Clear? Right. So that's it. We talked about fats. We talked about phospholipids. That's all you need to know. Right. Then functions of lipids. So we'll talk about the functions. Then we'll do some questions, and that's it. So your job is to study the highlighted parts. Study the highlighted parts. I'll be giving another essay question for you to write today as well. Okay. Then the marking scheme of that will be given. Got it? Functions of lipids. Please highlight. Okay. So I hope all of you have gotten an essay writing book, CR book. Okay. Food reserve as energy source. Okay. Your fats are a very good source of energy, but more than a source of energy, it's a good storage form. It's a good reserve because your fats you can store them for a long time, right? Everybody has fat under the skin, no? Do you know what they are called? They are called subcutaneous fat. Subcutaneous fat is found under the skin. It is good. It acts as an insulator. Okay, it acts as an insulator that is going to stop you from becoming too cold, maintain heat in your body. Okay, then what else? Maintain the fluidity of the plasma membrane, such as phospholipids and cholesterol. Can you see in this the model of the plasma membrane? I have put some cholesterol molecules here and there. Those cholesterol molecules prevent this plasma membrane from becoming too tightly packed, right? So these cholesterols help to maintain it as a fluid layer, maintain fluidity of the plasma membrane. Phospholipids and cholesterol act as signaling molecules, hormones that travel through the body. Okay, I want you to write down steroid hormones. Steroid hormones. Steroid hormones. So if you take hormones, hormones are mainly of two types. There are lipid-based hormones. There are protein-based hormones. Okay, so hormones, if you want, you can write. Okay, yeah, small chutiಯಲ್ಲಿ hormones. You can write small note. Two types. You have steroid hormones that are the lipid-based hormones. Then you have peptide hormones. Peptide, okay, peptide hormones. They are your protein hormones. Steroid hormones and peptide hormones. You can't read this. Steroid hormones. Katti not sleeping neither. Send me a response. Start practicing sitting. No. Okay. Forgive me for making sure you are getting the best maximum use. Okay. Right. Hormones are of two types, lipid based and protein based. Steroid hormones are the lipid based ones, peptide hormones are the protein based. So here, most of the hormones, as I mean, these hormones are steroid hormones. Can anyone give me an example for steroid hormones? Your sex hormones. You can write here sex hormones. Testosterone, estrogen, progesterone, steroid hormones. Sex hormones are steroid hormones. Found as a component of the animal cell membrane, cholesterol. You can highlight that as well. You can see here, cholesterol. Okay. Okay, these are the functions. Any questions? Any questions you want to clarify? I will show you some of the real life. I think I had before we move on to questions. Just to keep your interest, I think I had some. This is something I used for your original introduction, where I started this 2028 class. It's a bit of a big document, I think. Yeah, I have removed some of the things from this document. Yeah. So, can you see this is okay? I hope you won't be bothered by this. This is something we do in the first 2 years. That is called as doing dissections. Okay, so this is a leg. Okay, this is a leg, and the skin has been we have already removed the skin, and these are some of the muscles we have cut here and removed. So you can see some of the nerves. These are the sort of white color sort of strings like things are the nerves, and sort of tube like things, can you see sort of tubular things, those are the arteries. Okay, so this is something I photographed and I labeled, then I sent to my group means to the batch group, so that they can learn from what I identified on that day. So I go to the dissection hall, then I see some structures, then I refer my textbooks, I have some softwares also, then I identify them, then I label them, then I put them in the group so that others can learn from my work as well. So that's how it works in university. It's not very competitive like in A levels, because in the university, if everyone gets uh, marks above 75, everybody is going to get a distinction, right? But in A levels, the number of A passes is going to be limited, because why? They can only send a limited number of people to university, no? But everyone in the batch in university got excellent marks. Everyone is going to get a distinction. Okay. So this is what I want to show you is men may this yellow color. This is a skin. This is a skin piece of skin. Can you see the yellow color? That is the subcutaneous fat. This is the subcutaneous fat that is found under the skin. So that is a nice storage of energy that you can use when you are starving, not having eaten for a long time. Okay, subcutaneous fat. Right. Yeah, that's enough. Okay, we'll do one question from lipids quickly, because I need to cover proteins also. Do this part, these three, these are structured essay questions. Can you do these three? Adipose is the name of the tissue containing the stored fat. Adipose. When it's found under the skin, it is called subcutaneous fat. Subcutaneous means under the skin. Adipose is the tissue. Okay, muscle tissue. Okay, there's adipose tissue. Okay, let's check this. What is the name of 3 major types? Yes, I don't need to write, right? You have fats, phospholipids, and steroids. What is the type of lipid that forms a major component of the cell membrane? Yes, phospholipids. Yeah, phospholipids. They are asking major, right? You can have some cholesterol, but it's not a major, no. You can have some here and there, some cholesterol, but not major. No? Main structural difference between saturated fatty acids and unsaturated fatty acids. So you can say saturated fatty acids do not contain. Okay, I'll just write it in short form, right? You need to write in full. Saturated fatty acids do not contain double bonds. Unsaturated fatty acids can contain one or more double bonds. Can contain one or more double bonds. Okay. People tired? No, right? Okay. Shall we move on to proteins? Do these 2 questions as homework. Sorry, just one MCQ. Do this MCQ as homework. Which of the following statements regarding lipids is correct? Ah, no, okay. Come on, Damagano. Okay, do this. I'll give you 1 minute. Okay, so when we do questions in the class, you are allowed to look at the resource or the tute. Okay, during the class, you are allowed to look. But whenever you are looking up the answer, I want you to think about it. Think and refer. Okay. There's a question. Why can't we say about solid and liquid? Why? Why can't we talk about the solid and liquid? They're asking a structural difference. No, structural. I forgot to mention that. Mention the importance of that word. Structural difference means if you take a look at how the molecule is formed, then that's, I mean, that sort of a character is what you should write. If they ask a physical property, such as the solid, the physical state, then you can write liquid and solid story. But they're asking a structural difference in their composition, how they are made. Okay, so read the question carefully. Do this one quickly. Which of the following statements regarding lipids are correct? Send me answers, send me answers. Send me answers. Okay, that's the answer. Okay, very good, very good. Four. Good. Okay, lipids are macromolecules consisting of carbon, hydrogen, oxygen. No, right? They are not considered as macromolecules. Okay, nothing to write there. Each hydrocarbon chain of saturated fats contain one double bond. No. Okay. Do you need to write? If you like, you can write here. Saturated fats are composed of saturated fats are composed of saturated fatty acids. Saturated fats are composed of saturated fatty acids. Saturated fats are composed of saturated fatty acids. Saturated fatty acids do not contain, write down, saturated fatty acids do not contain, saturated fatty acids do not contain any double bonds. Saturated fatty acids do not contain any double bonds. Saturated fatty acids do not contain any double bonds. Saturated fatty acids do not contain any double bonds. Okay. When fats are formed, glycerol and fatty acids are joined by hydrogen bonds? No, it's a type of covalent bond. Can anyone tell me what is the bond? The specific name of the bond when glycerol and fatty acids are connected? Ester bonds. Hydrogen to oxygen ratio, you can write there. Make a cut karan, hydrogen bonds. Cut it with a different color pen, not black or blue. Okay. Hydrogen to oxygen ratio in lipids is higher than 2:1. Yes. Okay, you have much higher value of hydrogen. Why? You can see there's a hydrocarbon chain. No, hydrocarbon chain is carbon, hydrogen, carbon, hydrogen. You have only oxygens in the OH group of the glycerol and also a bit in the carboxyl group. Right? Nothing is nowhere else. Okay. Two phosphate groups. No, there's only one phosphate. Okay, nothing to be written. That is enough as reviews. You can cut the hydrogen bonds in the third statement. They are joined by covalent bonds that are called as ester bonds. Ester bonds. Had. Okay. Right. Let's go to talk about proteins. Let's go to talk about proteins. Very, very important topic. Okay. Right. Serious topic. Very important topic. Okay. Let's see it. Proteins are made up of amino acids. Proteins are made up of amino acids. That's really important. What is the building block of all proteins in this world? It's amino acids. Okay. 20 different amino acids. Highlight the number. 20 different amino acids are involved in formation of proteins. Okay. There are more than 20 amino acids in the world. If you take the group of molecules that are known as amino acids, there are more than 20 in the world. But out of those, only 20 are involved in making proteins. Only 20 are involved in making proteins. That's all you need to know. Okay. Elemental composition is carbon, hydrogen, oxygen, nitrogen, and sulfur. Can you highlight? There's always, usually, sulfur, but not most of the time. While you will see what are the exceptions. You will see what are the exceptions of where there is no sulfur, but anyway, it's very common. So that's we consider almost all proteins to have sulfur. Okay. Highlight the elemental composition. Okay. Right. At the center of an amino acid. So before we talk about the whole big picture of what proteins are, we need to know how they are built of. I mean, how they are composed of. So we need to know their building block. What is the building block? Amino acid. So we talk about the amino acid first. Okay. At the center of an amino acid is an asymmetric carbon highlight. There's an asymmetric carbon except in glycine. Glycine is a type of, it's just one type of amino acid. Okay. So glycine is a special man that I'll explain. Okay. Each amino acid composed of a amino group, a carboxyl group, hydrogen atom, and a variable group symbolized by R, which is an alkyl group. Alkyl is a organic you find in organic chemistry. It is used. It's a sort of a massive diverse group. Okay, you will see what we mean by alkyl groups. What we mean by alkyl groups. Okay. In case of glycine, R is replaced by a hydrogen atom. The R group is also called the side chain, where the other groups are the backbone. Hi. I know there's a lot of information there. I'll explain. Right. If you take an amino acid, the main man is the what we call as the center carbon. There's a carbon atom at the center. Okay. Then what are the other fixed groups? You have a carboxyl group, C double bond O OH. There's a double bond oxygen. Sorry, there's a double bond oxygen as well as a OH group. Then there's an amino group that we indicate like this, NH2 amino group. Then always in any amino acid you have a hydrogen atom. Then how the 20 different types of amino acids change from each other is by this one group. There is only one group that is changing the amino acids from each other. So the 20 amino acids differ from each other due to only one group that is called alkyl group. So let's label this. This is carboxyl group. This is the amino group. This is the variable alkyl group. This is the hydrogen atom. Everything you know. No? Then what I can do is I can sort of have a separation like this. All of these groups are called as the backbone. I'll explain why they are called as the backbone. All these groups are called as the backbone. And this guy, who is our alkyl group, he's called as the side chain. Okay, he's called as the side chain. Is called as the side chain. So you can see an amino acid is composed of 4 groups: amino group, carboxyl group, hydrogen atom, and an alkyl group. Then the amino, carbon, hydrogen, and carboxyl, they are all part of what? They are all called as the backbone. Backbone is what? What is our backbone? A vertebral column, no? Our backbone sort of supports our entire structure, no? It sort of holds our body straight, right? Provides support. Similarly, to a protein, this backbone set of molecules is the sort of the main core or the supportive structure. The side chains are sort of extending to the side. For this, you can see this diagram. Can you see this diagram? Here. Methane. This is an example of a sort of a long chain of amino acids. This is an example of a long chain of amino acids. Can you see? There's a purple color line. No? There's a purple color line. That is actually all the backbone molecules. What are the backbone molecules? We have carboxyl group, amino group, carbon atom, as well as the hydrogen atom. No? Then all the things that are extending out. Can you see all the groups that are extending out, such as what? All the groups that are extending out, such as, can you see this group, this group, this group? All these are side chains. All these are side chains. Got it? So those are the two parts of an amino acid. Got it? Those are the two parts of an amino acid. Then I'm going to draw our special man, that is glycine. Glycine is a special man. It's a special amino acid. Out of the 20 types of amino acids, glycine is one type of amino acid. There's also a central carbon. There's also a C double bond OH. There's also an NH2 group. There's also a hydrogen. Instead of this R group here, there's another hydrogen in glycine. Okay, so you call this is the glycine amino acid, which is the simplest amino acid. Glycine is the simplest amino acid that you have in your that's forming proteins. Okay, quickly take this down. Draw an amino acid. You should be able to draw an amino acid in your paper. If I am practicing you, if I am asking you to draw something in my tute, that is probably because it is going to be asked from you to draw in the paper. There are some diagrams in the paper, sorry, in the resource book that you don't need to be able to draw. But there are some diagrams that you might be asked to draw in the paper. Those diagrams I'm practicing you in the class. Okay. So, can you draw this amino acid? And on the side tute, you can draw the special amino acid, that is our glycine. Okay. Quickly draw this. Okay. Done? Yeah. Okay. Right. Then let's keep reading. Anyone who's still writing, hands up? Drawing? No, right? I want you to I want you to have a clear idea about the side chain and the backbone story. Okay. All these three groups are the backbone. Only the R group or the alkyl group is the side chain. Then let's keep reading. Amino acids may have one or more carboxyl group and amino groups. How is that possible? Because as the R group, you can have another carboxyl. There are amino acids like that. As the R group, you can have another amino group. So you can have two amino groups. Highlight, this is very important. Amino group has alkaline nature. Carboxyl group has acidic nature. Very important. This amino guy is basic in nature. Alkaline. Alkaline means what? Basic, no? Then the carboxyl group is acidic in nature. Carboxyl group is acidic in nature. Amino group, alkaline. Carboxyl group, acidic. That is very important. When both characteristics are found in one molecule, they are known as amphoteric molecules. Therefore, amino acids are amphoteric. That's an important word that you need to know. Amphotericity. Again, in chemistry, you will learn about this amphotericity when you are doing something called buffers. Amino acids are buffers. That means you can use amino acids to prevent excessive acid base changes. For example, imagine in your body, your body should have a constant pH. What do you mean by pH? The concentration of H plus ions. Yeah, your body's pH can become acidic or basic due to different conditions. Okay, your amino acids in your blood, you have amino acids in your blood from the food you eat, from the proteins you eat. Your amino acids in the blood help to maintain the pH value of your blood at a constant value. It doesn't allow it to change. Okay, so that is an important property given to amino acids due to its amphoteric nature. Because when, can you understand, if you have an acid in the medium, it will react with the alkaline amino group. If you add a base, it will react with the acidic carboxyl group. So it is able to neutralize both. That is called a buffering action. Okay, but for now, don't think about those. Just remember, amino acids are amphoteric. Now we move on to, so we have learned everything about to know about our building block. As usual, we are now going to move on to how amino acids connect to form proteins. That's what we are going to learn next. So again, we have a new type of bond to learn today. We have a new type of bond, peptide bond. We have a new type of bond called peptide bond to learn. Got it? Peptide bonding is what we are going to learn now. Two amino acids undergo a condensation reaction. Again, this is a condensation reaction, removing a water molecule from both and producing a bond that is known as a peptide. Okay, let's see how two amino acids join together by a peptide bond. Have the Kattiya listen. This is very important, peptide bond formation. Okay. Okay. So imagine you have two amino acids. You have two amino acids. This one C double bond OH. Let's say R1, alkyl group 1, there's a hydrogen, then there's a amino group. Then there's another amino acid, okay, that is like this. This is the amino group, carbon, hydrogen, C double bond OH, and we call it, it's R group R2, because there are two types of amino acids. There are two different amino acids. Okay, make a cap. Hydrogen OH. Now there's going to be a new peptide bond formation. Listen carefully. This OH is going to come from the carboxyl. Hydrogen is going to come from the amino acid. Okay. OH is going to come from the carboxyl group of this blue color amino acid, and the hydrogen is going to come from the amino group of the other amino acid. So you are going to have a new bond formation. There is a condensation or a dehydration reaction. Okay, what are you going to have? Two amino acids that are connected. So you have a carbon, hydrogen, amino group, and R1, and you have a carbon, double bond, oxygen. There's a now brand new bond here, and then you are going to have the other amino acid. Who is going to be here? The nitrogen, hydrogen, carbon, hydrogen, R2, C double bond, OH. Am I right? This is simple, very simple. And also, you're going to have what is produced. What else is produced here? What else is produced? Water. Label this bond peptide bond. And can you give me a name for this molecule that is composed of two amino acids? Can you give a name for this molecule that is composed of two amino acids? It is called a dipeptide. Two amino acids. It's called a dipeptide. Okay. So this is the process of connecting amino acids with each other so that you get a nice long chain of amino acids. That long chain of amino acid is known as a polypeptide chain. That is a chain of like in a necklace you have beads, no mehema mehema mehema. You have amino acids one after the other. That is called as a polypeptide chain. The polypeptide chain will form later after some modifications a nice working protein. Remember, proteins are working molecules. If you take carbohydrates, they are either energy or storage, right? If you take lipids, they are either structural or energy source. But remember, proteins are like machines. They can work, they can do things, they can take signals, they can act as enzymes, they can protect you, right? Proteins are like machines. They work in your body. They are your machines in the body. So they are very complicated structures. Very, very complicated. This is simply how you start to make them. This is the sort of the unmolded clay, right? You just have a polypeptide chain. You are going to do so many modifications to that. So you get the nice final protein. Okay, quickly write down how a peptide bond forms to form a between two amino acids. Quickly, quickly, quickly. We are running out of time. Any questions? You can ask anything. Okay. Okay. Right. I hope you are following with me. Okay. We are doing a lot of work, I know, but I know you can handle this, right? Simple organic chemistry concepts. We'll move on to levels of protein structure. I think that's all the time we have to discuss today. Levels of protein structure. I need uh, next week I'll discuss functions of proteins. Okay, so because I can't rush that, a lot of functions I need to explain. Okay, come to the levels of protein structure. Okay, I told you. Now imagine if you are baking a cake, if you are for your birthday your mother is baking a cake, okay, then what are the steps? You have to be the servant of the mother, ne? You have to bring all the ingredients: sugar, flour, eggs, baking powder, vanilla, whatever, icing sugar, whatever. Then those are your amino acids. Then your mother bakes the basic sort of the cake, no, using the butter, flour, eggs, and the what else? Some other sugar. You bake the cake. Then is it enough to be the birthday cake? No, you have to have multiple layers. Then you have to have icing on icing covering. Then you have to have icing flowers. Then you have to put your name. Then you have to put candles. That's how you get a nice final birthday cake, no? So it's like that. So up until now what we have made is just our basic cake. You haven't done decorated it or done nothing to it, so it's not very useful, right? It's just a cake. So that is what we are going to talk about. So what we just made is actually known as the primary structure. The primary structure, what is the definition? This is a definition, okay? Highlight. This is a definition, very important with your highlighter for definitions. Unique sequence of linearly arranged amino acids linked by peptide bonds. If they ask you what is the primary structure of proteins, you have to write this. Then I think in the last week I explained to you how do you identify keywords? You have to say it's a unique sequence here. Then it is linearly. Then you have to say it is bonded by peptide bonds. If you haven't written those, I can't give you. If you haven't written all those 3 points in a paper, I can't give you marks. Got it? Those are keywords because they are linear. Then the arrangement of amino acids one after the other. Right? This is an amino acid. This is an amino acid. These amino acids are arranged one after the other. So it's a unique sequence how they are connected. The 20 amino acids can be connected in so many different, orders. Okay. That is all about primers. This is like the simple baked cake, butter cake, like simple cake. Then for that, we can do so many modifications. Let's see. Secondary structure. Let's read. Primary structure, coils and folds of a highlight. Can you see? Primary structure of a single polypeptide chain. That means only one such chain, you coil it, fold it as a result of intramolecular. Right? I want you to write down here, within the molecule. Intramolecular means within the molecule. Intramolecular means within the molecule, hydrogen bonds. What do you mean by hydrogen bonds? That are forming due to a electronegativity difference. You can remember. Okay. Between what and what atoms? Oxygen atoms and the hydrogen atoms attached to the nitrogen atoms of the same polypeptide chain. There's some small part missing here. Can you correct it? There's a small part missing. Metenta, please, can you add oxygen atoms of the carboxyl group? Can you add oxygen atoms of the carboxyl group and the hydrogen atoms attached to the attached to the nitrogen atoms in a way? Please cut this to the amino groups. Okay, I'll explain. Can you correct it? Oxygen atoms of the carboxyl group and hydrogen atoms attached to the amino groups. That is where you get the hydrogen bond. So listen, listen. This is an example of a secondary structure. This is an example of a secondary structure. Can you pay attention? Okay, this is probably your nitrogen. Okay, why do I say that? Because this is the carbon. There's a double bond here. Can you, if I zoom in, this is probably carbon, this is oxygen, because can you see there's a double bond here, right? So this is probably nitrogen, this is hydrogen. Okay, so that is the place where you are going to get the hydrogen bond. This is the hydrogen bond. Between what and what? Oxygen atom of the carboxyl group and the hydrogen atom of the amino group. Those are the two atoms between which you are going to get the hydrogen bond. Okay. Visually remember. Visually remember. Got it? That's my best advice for you to study. Remember the diagrams I give in the tute. So those are given so that you can have a visual memory of where things are in different places of the syllabus. Okay. Otherwise, if the syllabus is massive, you are going to get lost in it. Okay. Bye, Karan, anyway. But I'm training you. Okay. Of the same polypeptide chain backbone, this is also important. It occurs within the backbone to form the secondary structure. There are two types of secondary structures that you need to know in your syllabus. This is called the beta pleated sheet. This is called the beta pleated sheet. This is called the alpha helix. Beta pleated sheet and alpha helix. Then you need to know examples of each. Keratin for alpha helix, beta pleated sheet, spider silk. So can you see this alpha helix is like a spiral. The alpha helix is like a spiral, as you can see. That's why we call it a helix. The beta pleated sheet is something like this. Okay, so if I take a piece of paper, beta pleated sheet is like this. Can you see? That is, the polypeptide chains are sort of folding on each other. Right? The polypeptide chains are sort of folding on each other like this. So this is one side, and then it bends and it goes down like this. So this is how the beta pleated sheet forms. So what can you say? So you know spider's silk is very stretchy. The Makudal Kaddu, must have seen the spider's silk is very stretchy. That is because these beta pleated sheets, this sort of layers, you can pull apart a little bit. They have this sort of spring-like nature, spring actually. Got it? So because of that, the beta pleated sheet forming the spider silk has a nice sort of a stretchy nature. Got it? I can say this is the beta pleated sheet as well. Oh, no. Like this. Okay. Got it? Remember that. Got it? I hope you understood. Then, so we have done two levels. So you have baked the cake in the primary structure. Now you have done maybe added multiple layers, up to 3-layered cake. You remember, you're not confused, right? I'm sort of, what do I call it? I'm equalizing final working protein to a nice birthday cake. Okay, so we are at the secondary structure. That means you have added multiple layers to the cake. Now, what do? We can do the final level, that is adding icing. You can add the icing and you can do the decoration. That is going to happen at the tertiary structure. That is going to happen at the tertiary structure. Okay, listen. Secondary polypeptide chain bends and folds extensively. Extensively, it will bend. Right? I want you to use this set of words to describe. It's a precise, compact, unique, functional 3D shape. Okay, from the following from the following interactions. What are this list of interactions I want you to know? Can you see this is a hydrogen bond here? This hydrogen and OH, there's a hydrogen bond. What else? Disulfide bonds. Disulfide bonds, sorry, disulfide bonds occur between two sulfur atoms. Can you see here? Disulfide bonds. What else? Van der Waal is actually, you will learn in chemistry. Van der Waal is also known as hydrophobic interactions. That's, they are actually not attractions, are they? They are actually repulsions. They don't like each other. You can write here hydrophobic interactions or Van der Waal interactions are repulsions. Can you write here? Please write down. They are repulsions. That's an interaction also, no? An interaction means what? Some sort of a association. So an interaction shouldn't always be a bond. It shouldn't always be a attraction. It can be a repulsion as well. Okay. So that is hydrophobic interactions, or you can call them as Van der Waal interactions. So those are the types of bonds you need to know forming the tertiary structure. Examples. So now you have, listen carefully, a functional protein, because you have added the layers, you have added icing and decorated it. So most of the enzymes, myoglobin, albumin. We'll talk about who these are, albumin, myoglobin, who these are. We'll talk about them next week. Okay. Okay. This is actually myoglobin, I think I have given here. Yeah, myoglobin protein. You can label him. Mainly multicolored dekina is myoglobin. Okay. I'll explain to you what is myoglobin next week. Okay. Finally, the final structure, quaternary structure. Quaternary structure, aggregation of two or more polypeptide chains involved in the formation of one functional protein. Now listen carefully. Okay. Up until the tertiary structure, you had only one chain, one polypeptide chain. Now you are going to connect two or more polypeptide chains. Now, what are polypeptide chains? They are amino acids connected one after the other. So imagine if a polypeptide chain is like a necklace or a pabaluma, beaded necklaces. So it's, if it's a beaded necklace, one polypeptide chain, then you are going to get several necklaces to form your fourth level quaternary structure. Then this is important. The separate chains now have a new name. They are called subunits. Highlight the protein subunits. They are held together by inter and intramolecular interactions. This is hemoglobin, which is a very important protein that is involved in mainly what? Oxygen transport in your body. Oxygen transport in your body, because oxygen atoms can bond like this: O2, 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 O2. These are red blood cells. You know these guys. Your red blood cells. Can you label this? These are red blood cells. Inside red blood cells, you have hemoglobin. When I label karga, this is collagen. Can anyone look and tell me how many protein subunits do you have in collagen? How many protein subunits do you have in collagen? How many protein subunits? Yes. Okay. Label him as collagen. Write down composed of three subunits. Here, you know, this is hemoglobin. You can write here, it's a metalloprotein. Metalloprotein found in red blood cells. What do I mean by a metalloprotein? Can you see this iron? Black color, can you see? There's an iron molecule in four iron molecules actually. So sorry, four ions, ferrous ions there are 4 ferrous ions in the hemoglobin molecule. Can you see here? 1, 2, 3, 4. Because a metal ion is incorporated into a protein, we call that a metalloprotein. A metalloprotein. Okay. So hemoglobin is a metalloprotein. You can write here. How many subunits can you count? How many subunits can you count in hemoglobin? Light blue color 2, dark blue color 2. Yes, you can say 4 subunits. 4 subunits. Okay. 4 subunits. Okay. Right. Then take out your essay writing books. Write down a homework question to be written. Homework essay we are going to write. Okay. We are going to write a homework essay. Let's write a homework essay question. I'll send the marking scheme, and okay, next week I'll explain how to write essay. Okay, no wait. Okay. Listen very briefly how to write an essay. Okay, I'll download. Did everybody get the water marking scheme? Did everybody get the water marking scheme? Okay, listen carefully. People who left earlier, if they are watching until this point, then good. Otherwise, you are going to miss how to write a proper essay. Okay. Give me 1 minute until I download the water marking scheme. Okay. So this is the brief. I'm going to very briefly. I'm going to tell you how to write an essay. I sent to all the groups now the PDF. I'll send. I'll send. Hold on. Okay. Listen very carefully. Essay question, you are going to be need to write 4 essay. Okay. Listen. You need to write 4 essays in the A level biology paper. Each essay is going to be given marks out of 150. 150 times 4, 600. 600 will be narrowed down into 30 marks. 30 marks out of the 100 marks in the paper is going to be from essays. Okay, 30 out of 100. Okay, one essay is given marks usually out of one essay question like this is going to be given marks out of 150. Okay, out of 150. Usually that the earlier style was 50 points were there multiplied by 3 to get 150. But now and with the recent trend is you are going to get around 38 marks that will be multiplied by 4 so that you get a maximum of 150. You don't give above more than 150. Okay. So that is the trend. So you need to write all the relevant points. This is an essay question. So you need to write it in full sentences. This is how I give marking schemes. This is so that all the points are numbered from 1 to point number. In this essay, there are 34 written points I want you to write. You don't put numbers in your writing essays. Do not put numbers. Okay, but write them as paragraphs, sentence after sentence, nicely. break paragraphs. So how do you break paragraphs? Listen, you can have in this what I say, this is the introduction. So this is one paragraph. Write one sentence after the other nicely as a paragraph. When I'm checking this, I'm going to check if you have this point in this sentence, then I'm going to give marks. Got it? So here, if I said it's these words are there, vital inorganic molecule, then I'm going to give this 1 mark. Then if you have written both vital chemical constituent and biological medium, you can get this. Then it's based on the chemical structure. If you have written, you're going to get 3rd marks. Then physical and chemical properties provide the ability to make the water so vital, 4 marks. Like that, that's how I'm going to mark your essay. So when I'm marking your essay, I check if these points are there. Then it's easier for me. So when I write essay marking schemes on the board, I write like this. Okay. Then the second paragraph should be nice, ideally about the first property, cohesive behavior. You write all the sentences under cohesive behavior, then you break a paragraph. Then you write about ability to moderate temperature. Sorry, write about ability to moderate temperature. Then you break a paragraph. Okay, like that you break paragraphs so that the marking examiner understands that this student has nicely written all the points so that it's easier for them and easier for you to write as well. Because you can see, okay, after you have written the essay, I have done this very nicely. I have written all the 4 essential properties of water. That is how you write essay. Break paragraphs, write in full sentences. Okay, cover all the points. Go through this marking scheme once more and write all the upcoming essays. Try to write the marking scheme. Try to write the marking scheme that you think, okay, this is going to be the marking scheme for this essay. Okay, everyone, write down the homework. Briefly describe. Write down. Okay. Briefly describe levels of protein structure. Briefly describe levels of protein structure. Hold on. Sorry. This is on part B. There's going to be a 2-part essay. Okay, sorry about that. It's going to be part B. Then part 1, describe formation of peptide bond. These are the 2 parts. Okay. Write down the parts of the 2 essay. So in this up to speed sessions, this is going to be a essay writing based workload as well. This is something I didn't do to my main theory class. We didn't write this many essays. Got it? Okay, we will conclude there for the day. So here I'm going to finish our second day of up to speed sessions of rapid catch up sessions. Okay, I hope you are taking the maximum benefit, complete the tutes. Okay, so I appreciate that you are joining with this with the intention of covering everything in a rapid way. It's the, I mean, then you should be joining the regular theory class as well. Then otherwise that theory class is going to go ahead. So we'll be starting photosynthesis this Thursday. We already started Saturday. Okay, so join, if you are Thursday batch, you can join with the Thursday class at 3:30. Saturday batch at 1:30. On Saturday we'll be doing photosynthesis day 2. Okay, right, that's it. And good night. Any questions if you have, you can ask. Yeah, I'll be adding to the group. Anyone else who's not in the group, you can send. Yeah, yeah. See you all at the classes. Yeah, I think, yeah. Everyone who's not in the groups, please send. Okay, good night, people. Good night. See you at the classes. Okay. Any issues, please contact the hotline numbers. They are ready to respond to any of your issues. Okay. Keep the groups active. Active means be aware of what I put in the groups. Okay. Okay. Good night. Good night.